So, yeah. So good morning, everybody. We're very happy to have you all here. So this is the first CannaLatan ISN, International Society for Neurochemistry School. It's been a long story to organize this initiative. This is a school that was planned 3 years ago, before the COVID thing came to our world. And so, I mean, it's been like a long time waiting and organizing, and a little bit of tension to make sure that you guys could travel around, with all the restrictions, that there would be no health issues, there would be no COVID. So finally, I mean, we're very happy that we are all here. Just to mention a little bit for those that are not aware, the CannaLatan thing is a research network who is organizing this activity together with the Complutense University and the University of Sao Paulo team. Okay? So this is a network which is funded by a Spanish and Ibero-American foundation, and we gather like 9 countries from Ibero-America, including Spain and Portugal, and 11 research groups. So the idea was to have educational activities in order to promote scientific training and promoting background and networking on cannabinoid research within Ibero-American countries. So this is kind of the focus, and to improve the quality of the science that we are doing and improving the applications and doing things better in general. So that's to say who's organizing this, why we're here. We have 3 days of contents. We divided the days. First day we will talk more on mechanisms of action and cannabinoid actions in general. Second day will be devoted to neuropsychiatric actions, so either the good and the bad things of cannabinoids, of course. And the third day will be devoted to the real world of patients and clinicians, so trying to see, after what we have seen in preclinical research and basic research, try to see what is real and what really can happen in the clinic. So this is the idea for the third day. Actually, it's a very timely event. In Spain, we are actually discussing for the potential regularization of medical cannabis use. Spain is like the Spanish paradox, I will say, for medical cannabis. We have some scientists. We have a Spanish Cannabinoid Society of Research, which is pretty potent and is very active. Patients and patients mainly are very organized around the Observatorio Español for the cannabis medicinal. And of course, there are companies also exporting already THC and CBD from Spain. However, there is no way to get some regularization of this. It's been very difficult in terms of political balance. They are very prudent. They really, it's like we are an island in the middle of Europe, and I would say in the middle of the world, actually. So they are not accepting the clinical evidence wherever they are, which are accepted in other countries. So this is the Spanish paradox, but we have to deal with that. Afternoons will be more devoted to practical sessions. We have also like 3 different experimental modules for the students that are participating in the school that we hope it will be interesting for all to learn the techniques. The people here in the Complutense University has done their best to have, to give you some hints and tools about what are the typical experiments that we do for cannabinoid research in the CNS. And we have also a session on medicinal chemistry on cannabinoids this afternoon, and tomorrow we will have a session on professional development. That's basically the content of the school. We have also launched, yesterday we had a very interesting session with the students. You guys were presenting your work, so we get to know a little bit what you want to do. And one of the ideas of the network, again, and I believe as scientists we should do that, is like try to promote your own creativity and ideas. So like we promote a context, an award for cannabinoid research. So we are offering you guys to try to work on your proposals, prepare like a short 3-4 pages proposal. We will have an award for the 3 best projects, and of course, if one of the projects is among the CannaLatan network, we can fund and stay and research and stay with that. So this is an idea. So you guys like take the initiative and the leading role on your thesis, on your postdocs, so on. And I think I'm basically done. I would like to thank all of you for coming here. I mean, I know it's an effort to travel from countries. It's been very difficult to organize the logistics, but you are all safe here. It's not as hot as we were worried last week, because last week was horrible. So I'm sure you will have a very pleasant stay. I hope you learn a lot, and the most important is that you make good friends and good connections, because that's one of the basics of good science, also like doing science with good quality of life as well. So thanks to all, thanks for the speakers and the faculty that have done the efforts to prepare the talks, and we can move on and start with Ken. Samia will introduce, sorry Samia, I didn't introduce you. Please. So Samia is my co-chair, I mean our co-chair for the school, and she will take the leading role now. Thank you.
Thank you, Ismael. I just have to actually say thank you for the opportunity to co-chair and invited me. It has been a great experience in many ways, and I'm happy to introduce our plenary speaker, or the speaker for our plenary talk, is Professor Ken Maki. I think he's very well known in the field, but I would just like to say a few words about him. He's a distinguished professor at Indiana University in Bloomington, and former director of the Gill Center for Biomolecular Science. And he has authored like more than 200 publications in this field with thousands and thousands of citations. So very well recognized, have made important contributions, for example, describing the distribution and signaling mechanisms of cannabinoid receptors, and also the identification, and especially for CB2 in DPR55, and also as part of the classification of the receptors at the International Union of Pharmacology. So I think this is just to make a long story short. So thank you very much for accepting our invitation, and of course enlightening the program of the school. So without further ado.
Well, thank you, Samia, for the kind introduction. Before I get started, I just wanted to say a few words. Organizing a school/meeting like this under the best of circumstances is really challenging, and I think we owe a great debt of gratitude to Ismail and Samia for organizing it under these conditions. I don't even want to know what they had to experience to go through this, but let's give them a little bit. Sorry. This worked when I tried it beforehand, but right now it's not.
He needs help with Spanish, I need help with the glasses.
But not the people on the internet. Okay, good. Again, thank you, Samia, for the kind introduction, and Ismail, Samia, for inviting me here today. I'd like to talk about a topic that is unpublished. It's work that we've been doing in the lab for the last few years, and hopefully we'll be publishing it soon. But it's a fun project to talk about, in part because the way it evolved, and as we all know, doing science is a lot of work, it's hard, time-consuming, takes a lot of time, but it's also fun, which is why a lot of us do it. So this was really merging of three different lines of research that kind of came together organically. The first is when we do a lot of developmental studies with THC, and one of the postdocs in the lab noticed when he was dosing mice with THC, he found a transient drop in body weight that then recovered pretty quickly. The second thing that was going on in the lab, totally different. We were screening orphan GPCRs to see which ones might be activated by THC, and of the receptors found, this orphan receptor GPR119 was activated. And then there's a large body of epidemiological data that has found that cannabis users are relatively metabolically protected despite consuming a not very healthy diet. And so the question is, how did we tie these three lines of research together? So that's the kind of general overview. This is the more technical overview. So what I'll do is give a brief background on endocannabinoids and the metabolic effects of chronic cannabis use. Then talk about our studies examining THC and signaling through GPR119, and specifically focusing on the effects of THC on high-fat diet-induced obesity, some data that THC is a GPR119 agonist, some more data showing that THC can increase secretion of incretins from intestinal enteroendocrine cells, and then ask the question, does THC decrease weight by a GPR119, and then talk a little bit about some preliminary metabolic effects of these results. So this is work that almost all was done by Amy Dopischwacker, who is a postdoc in the lab, and funded by NIH and NIDA, and we got cell lines from Dan Drucker and Richard Martin. So you'll see, the next few days, a lot about endocannabinoid system. So this is my brief way of conceptualizing it. It's a well-known neuromodulatory system, consists of endogenous cannabinoids such as 2-racanolglycerol, anandamide. Generally made, endocannabinoids are made and released following increases in either neuronal activity or other cell activity, either by changes in membrane potential, as debolarizing the membrane, increase intracellular calcium, or activating phospholipase C-beta. They're synthesizing enzymes such as deacetylcholipases, NAPLD, several others, degrading enzymes such as FAW, maglipase, ABDA6, ABDA6, and then receptors like CB1, CB2. So triangle lipids, giving endocannabinoids, being metabolized and reincorporated into membrane lipids, and acting at CB1, CB2, and other receptors embedded within membrane lipids. So that's kind of the conventional view of endocannabinoid system is I think, a very useful way of conceptualizing it. However, like everything, once you get into it, it's much more complicated. So in general principles, lipid signaling and their targets are incredibly promiscuous to someone who kind of grew up with catecholamines and things like that. So I talked about the N-acetylethanolamides like anandamide, acetylglycerols like 2-AG. However, on the ligand side, there are the taurine compounds, so lipoamino acid, prostamides, various cyclooxygenase metabolites, either cyclooxygenase or others. And then on the ligand side, the receptor side, there are multiple G protein coupled receptors such as CB1, CB2, but also GPR55, GPR18, etc. Various of the TRP-V channels, the TRP-M channels, some ligand-gated ion channels, as well as nuclear receptors. So if you think of 2-AG, CB1, CB2, just the tip of the iceberg, and there's a whole lot more under the water that we should at least be aware of when we're working with these molecules. Okay, so to move on to cannabis use and metabolic risk. So activation of CB1 receptors, well known to stimulate consumption of highly palatable foods, such as Harold and Kumar, very strongly driven, lots of motivation to get to White Castle for their burgers. And in rodent models, feeding small amounts of THC orally lead to increased food consumption over a period of time. And this is the food consumption portion of this process is essentially driven, so injections of small quantities of 2-AG into nucleus accumbens increases food intake. If you block CB1 receptors, this is using the Merck compound Tamaranban, you see an increase in energy expenditure, and most famously work from Sanofi in trying to develop the weight loss drug Rimonabant. Blockade of CB1 receptors decreases body weight and improves several measures in metabolism. And this ran through many subjects. And this is the essential data there. So 20 milligrams dose leads to rapid and sustained weight loss that's reversible if you stop taking the drug. So that's a little puzzling. Those data make this following observation a little bit puzzling. So with chronic cannabis users, despite an increased caloric consumption of 200 to 500 kilocalories more than weight-matched subjects, several, but not all, so about 90% of the studies come out with this conclusion, is that chronic cannabis consumption may have beneficial metabolic effects. So decreased body mass index, decreased waist circumference, decreased risk for metabolic syndrome, decreased insulin resistance, decreased risk for type 2 diabetes, and decreased C-reactive protein. I mean, that's a pretty good profile for a drug in at least in American society, where we have a really big problem with obesity. But this is puzzling given what we know about CB1 receptors. So these are some of the data. There are a variety of studies like this, but one I chose. So you can see, and this is divided into individuals who never used cannabis, had a significant past use history, and are using currently. And the red box parameters are the ones that I want to draw your attention to. So decrease circulating insulin levels, increase or improve less insulin resistance, lower BMI and lower waist circumference. Lower waist circumference is a really good measure for kind of visceral fat, which is the bad kind of fat. Interestingly, there seems to be a bit of a sex difference to that. So males in both the waist circumference and the HDL cholesterol numbers seem to be better protected than females. Okay, so that's kind of epidemiological data. So how do we examine that in a preclinical model? So what we did was we took wild-type CPT7 mice of both sexes and started them young for a variety of reasons, partly economical and partly because that's in the U.S. when poor diet starts, and fed them 3 months of a research diet, 12331, which has this composition of protein, carbs, and fat, and most of the fat is the bad partially hydrogenated coconut oil fat, and let them grow up till they're about 40 grams, so reasonably obese for a C57 mouse. And it takes about 3 months to do that, to get them obese, but that's kind of what they look like too. They are quite big compared to our usual C57s. So in this experiment, we gave these obese mice either vehicle, 2 doses of THC, or a known positive control, in this case a PPR1 amine agonist, all IP daily. And this is what we see. These are the vehicle mice. They continue to slowly gain weight, but 3 milligrams per kilogram and 10 milligrams per kilogram mice lose weight, and comparable, 10 milligrams per kilogram is comparable to what you see with the authentic weight loss movement. So, this work suggests that daily THC given to an obese mice causes weight loss. As I touched on, the role of CB1 in metabolism has been extensively explored, where CB1 agonism promotes weight gain, CB1 antagonism promotes weight loss. So we don't think THC is causing this effect via CB1 receptors. The role of CB2 in metabolism is less clear, but appears modest. But just to keep in back your mind, CB2 receptors are very involved in inflammatory responses, and inflammation is a prominent component of obesity. So this led us to think that THC might be acting on other targets. So this is presented as a logical path that we did this and this, but of course these are 2 independent things going on in the lab that just sort of converge. So we're examining candidate receptors by using receptor internalization as a surrogate for receptor activation. So we're going, the previous data is in mice, and now we're moving to cells. So many of you who work with G-protein coupler receptors know well that many G-protein coupler receptors live happily on the cell surface of cultured cells or other cells. And if you treat them with an agonist, they internalize rapidly. We looked at this for CB1 receptor quite some time ago, and we're quite interested in various steps involved in the process. And while these pictures are very nice to show in a talk, they're very challenging to quantify. So Jim Wagermiller, my lab manager, developed this quantitative internalization assay quite some time ago now, and it's like any nice technique. It's really simple, so you can do it very, very reliably. So grow receptor expressing cells on a 96 or 3-4 well plate, then stimulate with drugs, with or without protein synthesis inhibition, depending on the nature of the experiment, and then fix in 4% paraformaldehyde. And under our conditions, 4% paraformaldehyde does not permeabilize the cell. It just fixes it. And then since these cells are non-permeabilized, if you incubate with an aminoterminous antibody, because these are GPCRs, so the aminoterminous is outside the cell, you'll just label receptors still on the cell surface. We typically, in transfected cells, do this with the HA or other epitope tag, but we've done this with native cells, so CB1 receptors make good antibodies against the aminoterminous, so you can look at this process in cultured neurons as well. And then detect with a near-infrared fluorescent antibody, and that's the key to this technique. It really decreases the noise of the background fluorescence, so it increases your signal-noise ratio. And we use light for Odyssey, but you can use any infrared imager, and then normalize the cell number. And if you just do a scan of the plate, this is what it looks like. So this is CB1 receptors here treated either with this synthetic cannabinoid, this spice compound, or the aminoalkyl phenolwin 5.212-2. And for increasing durations of incubation, you can see that as the receptor internalizes, the amount of receptor you can detect on the cell surface goes down. This is very easy to quantify, and you can do lots of replicates, fairly confident estimations of the numbers. And you can do neat things like look at how fast receptors internalize, things like that. And so this assay gives you surface receptors at a specific time. As an assay, it's nice. It's extremely flexible. I'm showing you receptors going in from the cell surface, but you can run the same assay to look at receptors going out the cell surface and things like that. So we did that for a number of G-protein-coupled receptors, including GPR119, and this is work from Johnny Latham, he's a student in the lab with Jim. For GPR119, 2-oleoglycerol is one of the endogenous ligands for this receptor, so that's our positive control. THC, of course, and we have to look at CBD in most assays we do because there's so much interest in CBD, so we ran that as well. So you can see that both 2-oleoglycerol and THC internalize the receptor with moderate potency. So that, of course, asks, what is GPR119? So it's an interesting GPCR, couples both the GS and GQ, so increase cyclic AMP and increase COX-2 metabolism. It's expressed at high levels in pancreatic islets and in some gut endoendocrine cells, so the K and L cells. So this is work from a company that was investigating this receptor, so staining of islets, but looking at the particular subtype of cell seen in islets in rodents at least are beta cells, and then looking in intestinal villi, you can see the endoendocrine cells in the crypts or label. So as I mentioned, 2-OG and also oleoethanolamine are proposed endogenous ligands for this receptor, and the idea is these ligands are produced when fats are broken down, and this is detected by this GPCR that then changes cell physiology. Activation of GPR119 increases glucose-sensitive insulin secretion in islets by cyclic AMP, and then incretin secretion into endocrine cells with things like GLP-1, GIP, UIY, etc. So really a receptor that's very well positioned metabolically to do interesting things. And there's a number of studies that GPR119 activation is metabolically beneficial, at least in rodents. So this is just another model of endoendocrine cell signaling. So this would be THC interacting with the receptor, and then leading to secretion of these peptides, of the incretin peptides, that then may interact locally with afferents from vagus. These are primarily active in humans in small intestine, particularly in duodenum and jejunum, and eventually also make it out in the circulation where they circulate more widely. So we showed that THC activated internalization of GPR119. So we're interested in some of the other signaling pathways that GPR119 uses. So we looked at cyclic AMP using time-resolved fluorescence, IP1 using a homogeneous FRET assay as a measure of PIP2 hydrolysis, and then phospho-ERK in cell western, and then internalization, which we already did. So THC at GPR119 stimulates internalization. That's the assay that you saw before, and also activates IP1 production. So stimulates PIP2 hydrolysis to generate IP3 and diacylglycerol, and CBD is not. But when we looked at ERK and cyclic AMP stimulation, you can see that THC doesn't stimulate ERK phosphorylation unlike 2-OG, and also doesn't stimulate adenyl cyclase through cyclic AMP. So THC is a pretty strongly biased agonist at GPR19, at least in these cells, favoring GQ signaling over GS signaling. Okay, so earlier I told you that GPR19 increases insulin secretion via increases cyclic AMP in beta cells. So from what I just showed you in HEC cells, we wouldn't expect THC to increase insulin secretion. So we looked at that. So we used Min6 cells, which express GPR19. They also express CB1 receptors. And then looked at high glucose to simulate insulin secretion, so GSIS. And this is normal. This is basal insulin secretion. You increase glucose to about 17 millimolar. You see increase in insulin secretion, which is what beta cells normally do. If you also add a balanced GPR118 agonist, the AR compound, you see a further increase in insulin secretion. However, THC does not increase insulin secretion. And you can think this is probably good in a case where you already have insulin resistance. Increasing insulin secretion isn't necessarily that beneficial for the organism. Not shown here, but if you activate CB1 with a potent synthetic agonist, you do increase GSIS. Okay, so so far I hope I convinced you that GPR19 is activated by THC. However, those of you who kind of work in the field have realized that we're using micromolar concentrations of THC, and there's this little kind of sticking point that this is where primarily twistis. A human smoking cannabis cigarette, may get transiently peak levels of 500 nanomolar or so of THC. And we really, to get good GPR19 activation, we need levels around the micromolar or so. So how can we solve this problem? So what about THC metabolites? These are plasma levels of THC, but we're really focusing on GPR19 in endoendocrine cells in the gut, particularly in duodenum and the jejunum, which will be subject to bile and any THC metabolites in the bile. So as many of you know, THC is metabolized phase 1 metabolism, cytochrome P450s, particularly CYP2C9, and then is glucuronidated. This is a well-known active metabolite, probably slightly more potent than THC, and this is a classic inactive metabolite that's excreted either this way or after glucuronidation. So this is an interesting study that looked at concentration of cannabinoids and THC and its metabolites in bile at autopsy from individuals who were using cannabis. And the number that jumps out at you, one is the carboxy metabolite is almost a micromolar, about 800 nanomolar, and the glucuronide is about 18 micromolar. So really, really high concentrations of these in the bile. So we then looked at the effect of these metabolites on GPR119, and the carboxy metabolite internalizes the receptor reasonably well, as does the glucuronide. The MBX compound is another synthetic GPR119 ligand. Stimulates the adenylyl cyclase, as well as IP1. Interestingly, these both of the metabolites strongly suppress ERK1, ERK2 phosphorylation, so leading to dephosphorylation of the kinase. So that's the signaling. We're at, what endocrine cells do. They secrete incretins. So we're interested in how do these metabolites work at incretin secretion. So for this, we got the glutag cell lines. These are SP40 transformed mouse L cell made by Dan Drucker many years ago, and they retain many of the features of L cells, which otherwise are difficult to work with because they're like 1 in 1,000 among all the other cells in the intestine. So again, THC stimulates GLP-1 secretion from these cells, as well as THC metabolites. So at this point, this is the working model that we have, is that THC and its metabolites in the gut will stimulate GPR119, probably through phospholipase C, increase intracellular calcium, lead to incretin secretion such as GLP-1, and then all the good things that GLP-1 and other incretins do. So that's a nice model, but that doesn't really explain what's going on. So really the key question is to move back from cells to mice now. Is GPR119 needed for THC-induced weight loss? So to do that, we use the same model again, except this time we're also looking at GPR119 knockout mice. This is the data I showed you before with weight loss with the wild types. And in GPR119 knockouts, the weight loss is strongly attenuated. So we wanted to make sure that these mice could actually lose weight, so we used our other favorite compound for weight loss, rimonabant, and you can see that 3 mg per kilogram rimonabant causes rapid weight loss in these mice. So, everything I've showed you up to now has been IP injection of cannabinoids, of THC. But thinking about this, we're really focusing on the metabolites, and the way you increase metabolites of THC is to feed the THC orally. So, presumably oral THC will be more effective due to first pass metabolism because the oral bioavailability of THC is pretty low because it's metabolized so rapidly in the liver. So what we did was to feed the mice THC or vehicle and sweetened condensed milk, and see if this works. So this is just a P200 pipette tip cut off filled with sweetened condensed milk with a drug or vehicle. Just bring it down to the mouse, which is gently wrapped in a paper towel. They love it they love it a whole lot more than getting daily IP injections. So if we do that, we gave doses ranging from 10 to 30 milligrams per kilogram, and you can see that you see progressive weight loss in a dose-dependent fashion, which we find is quite interesting. Okay, so just as a control, if you give an oral GPR119 agonist, you see a similar effect, although it's a little bit delayed. And oral and the synthetic GPR119 agonists are ineffective in the GPR119 knockouts. And then also, we want to see how reversible is this process. So again, the diet-induced obese mice, treat with THC with 2 weeks, then stop, and then maintain a high-fat diet throughout. And sure enough, they lose weight. The amount of weight they lose plateaus, but then if you stop the drug, they rapidly gain weight back again. So it's quite reversible in the way that we're doing this here. Okay, so what I've shown you is that THC can cause weight loss in obese mice in a GPR119 dependent fashion. However, that's not really the epidemiological situation. In humans, it's many years of high-fat diet plus THC or plus cannabis use. So you're really looking at prevention of weight gain rather than weight loss. So we wanted to look into that. Oh, I'm sorry, got ahead of myself. One thing quickly before that is that the chronic cannabis use is associated with decreased risk for type 2 diabetes. So we're doing oral THC treatment, then looking at glucose tolerance after a 1 gram per kilogram glucose load, and you can see that in obese mice, 2 weeks of oral THC treatment improve glucose tolerance in both males and females. Okay, so now this is the epidemiological experiment. So the question is, will THC prevent weight gain from high-fat diet? So this is work done by Parsh Kumar, who was an undergraduate in the lab, has stayed on as a post-baccalaureate student in the lab, which kind of gives you a sense of how long these experiments take to do. So he started with 35-day-old mice, fed them a high-fat diet, co-treated with THC at various doses. First I'll show you 10 mg per kilogram, orally 5 days a week. So we didn't want to do it 7 days a week because these are essentially 2-3 months experiments, and didn't want either Parsh to have to come in every single day of the week for that long, or the mice to be handled that much. So sort of like the reverse party protocol. So here You can see that in this model, THC decreases weight gain. And if you break out the males and females, the effect may, in this particular experiment, may be more in the males than females, although I don't think that's going to hold up. And then, so thinking a little bit more about this, generally the way it works is people have a poor diet first, and then they start cannabis. So instead of the previous experiment, you started cannabis simultaneously with a high-fat diet. This is starting poor diet at 6 weeks, and then start THC at 2 weeks, and then co-treat with THC 20 mg per kilogram 5 days a week. And you can see in this case a decrease in weight gain from the mice receiving THC, both in males and females. So it seems is a pretty robust finding. Okay, and then finally just wrap up with some preliminary data. In looking at, this is Brush's model after 6 weeks of THC, looking at liver triglycerides, you can see that THC decreases triglycerides as well as lipid staining by percent area in the males, but interestingly doesn't have any effect in the females. This sort of harkens back to epidemiological data where males seem more responsive to THC than females. Okay, so that's the last experimental data I want to show you. If I have a few minutes, there's some summary stuff I want to go over because there's, I think, some interesting things here in terms of how we do science and how we think about what other people's science is like. So, you know, what I've shown you so far, I hope convinces you that GPR119 activation could be beneficial with weight loss. Working in GPCRs, nothing you do is ever original. So someone has done something similar, at least with another GPCR, or your GPCR in another context. So GPR118, when it was cloned, the distribution was like, you know, we need to look at this for metabolic disease things. So it attracted much interest in the pharmaceutical industry as a treatment for type 2 diabetes, as a potential treatment for type 2 diabetes. So most clinical studies had metabolic parameters like insulin, HbA1c, et cetera, as their primary endpoints. And so what are the results? This is too small to read, but you don't have to read it. Essentially, 9 failed phase 2 clinical trials with GPR119 agonists for type 2 diabetes. Essentially not showing good efficacy. And why might that be? So development of GPR119 agonists is focused on the following. You know, synthesizing agonists that increase cyclic AMP and increase glucose-stimulated insulin release, so really the blogging the dying beta cell approach to treating type 2 diabetes, to maximize oral bioavailability because they want an orally active drug. So they've selected for GPR119 agonists that may not necessarily stimulate GQ, PLC, and calcium increases. Ideally, we'll have low concentrations of gut lumen because you want it to be absorbed, well absorbed into the body. And there's work that beta cell GPR119 is not even needed for glucoregulation by GPR119 agonists. That's work from Dan Drucker's lab a couple of years ago now, where they conditionally deleted GPR119 from beta cells and showed that treating GPR119 agonists increases normalized glucoregulation quite well. And then there's some data from human pancreatic RNA-seq studies suggesting that actually in human pancreas, GPR119 is expressed in alpha cells, which of course secrete glucagon and is not necessarily what you want for treating type 2 diabetes because of increased blood glucose. So, so that's kind of how we're reconciling our results with what's out there in the literature. So THC causes weight loss in these mice and lessens high-fat diet weight gain and improves glucose tolerance likely by GPR119, although we still need to show the latter in the GPR119 knockouts. So the general hypothesis is that THC and metabolites, particularly carboxy metabolite and glucuronide, activate L-cell GPR119 to favor GPL1 incretin secretion over insulin secretion, and so it's metabolically protective despite a suboptimal diet in chronic cannabis users. The rationale is that THC activation doesn't increase cyclic AMP, which is needed for glucose-stimulated insulin secretion, and we found that THC doesn't increase glucose-stimulated insulin secretion. Question, does increasing L-cell IP3 and calcium favor GLP-1 and PYY secretion? That's not known. And sort of philosophically, is increasing incretin secretion better than driving beta cells to release insulin in a pre-diabetic state that's characterized by insulin resistance? And then, is it beneficial to restrict GPR19 agonists to the gut? Because of the systemic GPR19 agonists haven't worked very well. And then the future directions that we're going with this study is to follow up on the sex differences with the liver triglycerides, for example. We actually have some very interesting data on this that's not quite ready for prime time, but there's the question of bacterial metabolism by THC, and then also just general bacterial metabolites acting on enteric nervous system GPR19. With the collaborator, we found that when we give this oral THC, we see very profound changes in gut microbiome within 2 days. It's quite in a favorable direction. We'd like to see if we can actually see changes in incretin secretion in vivo. We have a history in the lab of investigating functional selectivity in other cannabinoid receptors like CB2, so we're interested that's really important for this receptor. And then, of course, what metabolic mechanisms underlie weight loss with THC? Is it decreased food consumption or increased energy expenditure like with the monobond? And then the durability of metabolic effects, because we saw rapid reversal of weight loss when in the acute diet-induced obesity experiments. But the human data suggests that past cannabis use may convey residual protective properties, and we'd like to look at that in our mice. And then there's kind of classic things like body composition, the brown and white adipose tissue ratios, uncoupling protein 1 expression, things like that after THC. And then finally, things if you combine a moderate dose of THC plus GLP-1, you see actually synergistic benefits, because some of these metabolic kind of dual approaches can be extremely synergistic. So with that, that's all I have. I'd like to thank you for your attention. Be happy to answer questions as time allows.
Thank you very much. We do have, it was a great talk, and we do have time for questions. And then I would like just to remind that we would give priority for the students to question initially, and we would really like the students to participate and engage in the discussion and questioning. And then we also always have time in the coffee break to interact more and ask questions, so everyone has time for that. But initially, we would like to invite the students to start with their doubts. If anyone would like to be the very first one. Yes. Anyone? Yes, I have prizes for the students that will ask the most clever. Yes. Please? Yes. Adriano. Then just when you ask, say your name, then everyone can know, also the speaker, who you are.
Thank you, Professor, by the amazing lecture. My name is Adriano Chaves. I'm from Brazil, working with Professor Samia. And I'm thinking about a very basic question. There is evidence about the contribution of GPR 119 for THC effects in CNS, in central nervous system. There is the evidence of contribution of GPR 119 for THC effects in CNS.
If I understood correctly.
Thanks for the question. It's interesting. There's actually almost no GPR119 in the central nervous system, at least by RNA-seq studies. there's a moderate amount in the enteric nervous system, but not in the central nervous system.
Yes, we help.
Hello, Professor. My name is Gunther van der Walt, and just once again, thank you for an amazing lecture. I really like the way that you progress with every subsequent study and how it builds on each other. It's quite a prime example for students in the field. But from that, I just have two basic questions based on what you left off with. I'm very curious about what type of changes in the microbiome that they have seen with THC administration. And then in a second part question coming from a metabolic background, is there any studies on differing lipid metabolism in any cells? So, specifically, most of my research is in the nervous system, but so yes, is there anything specifically related to the lipid metabolism in specific cell models that they've seen so far?
I'll answer your second question while I try. Okay. I'm sorry. I'll answer your first question now that let me show you, because I'm not going to remember and pronounce correctly the different bacterial families that are expressed, that change, but let me show it to you after the session. We haven't looked at specific lipid metabolism within in these mice, and it's a very interesting question because obesity is accompanied by pretty substantial lipid changes, and we don't know if those are normalized or not with GPR19 agonist treatments.
Thank you, Professor, for the amazing talk again. My name is Franciele, and I was wondering if you have ever seen if THC alters the levels of the endogenous ligands for the GPR119 receptor.
We haven't looked at that. It's an interesting question. The gut levels are dependent on diet, but whether we change circulating OEA or 2OG levels, we haven't tried that. It'd be interesting because THC does do that to endogenous cannabinoids.
Hi, Dr. Meki. So, just out of curiosity, I saw that male seems to have a more pronounced effect in terms of GPR 119, and I wonder if if the effect on females could be somehow improved when combining physical exercise or other kind of activity that can kind of, I don't know, somehow boost the effect on THC and GPR 119.
We haven't looked at that. It's an interesting question. For us, the female is more variable because sometimes it loses much weight as the males, and other times they don't, which makes it difficult to study because you don't know which group you're in. But it's an interesting question, and I think a lot of this is, if you think of application, real-world applications of any of these types of treatments, sort of the combination often works better than just relying on a single drug to do the job.
Any more questions? Okay.
Hello, Professor. Thank you for the great lecture. My name is Melissa Ribeiro. I'm a Brazilian. And thinking about that, normally people use cannabis in the context of kind of escape from stressful situations. And what do you think about the role of THC on the metabolic effects but under stressful conditions in these mice.
That would be a really interesting experiment to do. We haven't. I mean, just a little bit. I know stressed mice eat very differently. They're not very healthy. It would be there'd be a fun experiment to do.
Hello, my name is Luzia. I'm from Brazil also. And I was thinking about, besides the THC and CBD, your lab did work with other phytocannabinoids in this interaction with the GPR 119?
That's another interesting question. We haven't tried other compounds other than CBD, which doesn't work. Some like CBN or CBGA. Also, as far as I know, we haven't tried the acidic form of THC. We haven't tried THC yet. Any interesting study always opens up all the additional questions that one should look at.
So I think we have time for one last question, and then we go for the... Yes.
Thank you very much. Fantastic presentation, and we are learning a lot. I'm particularly interested in obesity, treatment with cannabinoids of obesity. But what we are seeing in the clinic is that our patients, for example, treated by with THC for pain, chronic pain, females are gaining weight, not losing weight. And you have shown that maybe differences between male and female regarding GPR 119. What would be the interpretation for these situations? We have included many of the women are abandoning treatment with THC, even they are doing better, fibromyalgia and other chronic pain, because they are gaining 3 kilos in 2 months, for example. So I would like to understand and how we can move forward from a clinical perspective with these interpretations.
Yeah, no, that's interesting. I hadn't heard that observation before on the weight gain with pain. Because I'm a little, intuitively you think it the other way around, less pain, maybe more physical activity than actually weight loss, but that's not the clinical experience.
But yes, but it's not necessarily compensated by the increase in activity. And also we see that with CBD, we have seen frequently weight loss and decrease in appetite, even in kids treated for epilepsy, for example. We have to check the weight in kids because they weight lost. They lost weight with CBD, and that's a little confusing, but for me.
And of course, we're taking the very reductionist approach here for how we're doing this. They're obese mice, but they don't have anything else going on with them.
Because it's also true that male that use cannabis for adult use purposes, they are not very fat. That's also true.
So there may be so in the clinic with your male pain patients using cannabis for pain, do they also gain weight, or is it more female? Is it restricted to and pre or postmenopausal make a difference? Does premenopausal or postmenopausal make a difference? Both equally. So. But it's like quitting smoking and then gaining weight. The trade-off is. No, that's very interesting. We should chat.
So again, I would like to thank you, Ken, for this very interesting talk. And then we have time now for the coffee break, and I think everyone is welcome to further discuss and ask questions, because I'm very sure everyone has many interesting questions that we can continue in the coffee break. And then we come back here for the continuation of the workshop or the course with 3 other lectures. Thank you, and I think then it's time for
So before we start, when we close the session at 1:30 for lunch is going to be in a different building. We like you guys to do this is like a white maze thing. So you need to find the food. So food will be in the School of Philosophy. You have the link in the electronic program that we send you. You have the location of the School of Philosophy. For those that do not use gadgets, it's when you come outside of the building on the main door, you take the big avenue. It will be sunny but not as hot as yesterday, and walk on the right-hand side until the end of the avenue. And then it will be a building on your left-hand side. It will be easy. The School of Philosophy. That's for the lunch time. And after that, and once we close this session on cannabinoid actions, we will also put the one slide with the distribution of the students for the practical modules this afternoon. So that we will do after the last talk this morning. That's it.
So that's our next session. That will be on cannabinoid actions in the nervous system, and I'm very happy to introduce the three next speakers. And the first one is Professor Hui-Chen Lu, and she's a professor at Indiana University in Bloomington, and currently the director, right, of the Gill Center for Biomolecular Science, and I think she will tell much more about her research today. Thank you again for accepting our invitation.
Thank you, and thanks Ismail and Samia for this wonderful organization and invitation for me to be here to share you our work and also to get you a bit more familiar with brain development. So I think I need this. So first, I'd like to advertise that the Gill Center is becoming a cannabinoid research center. And as you can see, Ken, who just gave the talk, and Heather Bradshaw, many of you probably also know, she is very good in majoring cannabis, endogenous versus phytocannabinoids. And we also have Isban Katona and Norbert Hills and many others. And so this work is teamwork. As you can see here, people involved found 3 different laboratories. My laboratory, Gabriel, who is right here, and Izaki, who has been back to Brazil, so both are Brazilian, and Heather and Claire, and Ken's lab, Maggie, who contributed quite a bit for the top work I will showcase today to you. And if any of you are interested in working on cannabinoids and interested to come to a beautiful, colorful site, please contact me or Ken. So we'll be happy to talk to you more about it. Okay, so, let me move. And so I don't know what type of brains you think you may have. Okay, so we all have different kind of perception about who we are. And I think most of us probably don't want to be like this. So the question is, how does this endogenous cannabinoid system contribute to this different type of brains that you may have? And so Ken already did a beautiful job of introducing endogenous cannabinoid system. I will just briefly reiterate a little bit. So the thing you need to remember, of course, endogenous cannabinoid 2-HG and anandamide, and it gets synthesized and degraded and act on the receptors. And so I want to have a brief introduction about this ugly neuron here. So basically, you have axon transmit the signal, connect to the postsynaptic partners through their dendritic compartment here, and so this is presynaptic compartment to the postsynaptic compartment. And so majority of our knowledge about how endogenous cannabinoid system work more for the mature synaptic system, I mean mature synapse and nervous system. So as you can see here, there are 2 types of synaptic connections, GABAergic and glutamatergic. So inhibitory, excitatory, and there are more prominent expression of CB1 receptor, the most prominent cannabinoid receptor and also the most dominant GPCR, G protein coupled receptor in the brain. It's enriched in GABAergic terminals here, and 2-AG can be uptake and regulate the CB1 receptor function, and then inhibit neurotransmission and get degraded by the enzyme. So, so basically, most of the time, endogenous cannabinoids is act in demand as a retrograde messenger to the presynaptic partner. However, whether this is true in the developing brain remain more studies to understand. So, and as many of you know, cannabis are getting extremely popular, especially among young adults. So you could see here, this is the most recent data. It's about 15.5% has experienced cannabis, and they get packaged in a very, very beautiful, like candies or cookies type of package. You don't really feel like they're anything different, and so you could have like strawberry flavor or peach flavor, whatever means the ratio of THC versus CBD, and so when I took this picture myself when I visited my son in San Francisco, I told him I want to go to dispensary, and he was like, "What am I doing, taking your mom to a dispensary?" He was very, very embarrassed. So anyway, and so you could see that in this popularity, there will be young women happen to consume cannabis, and with or without the knowledge of pregnancy, and it's accepted in a way that it's probably okay, it's natural products, so it's no harm. And so there are a lot of potential exposure, and of course you all know about CBD, and it has been extremely popular. You see every corner, and this is just right next to my hotel here. And so I assume this is Madrid specialized CBD. And so you could see like it has been advertised as a safe alternative for morning sickness or discomfort, and it's safer than any medicine you could take, which I'm not sure if it's true. We really don't know enough about CBD for nervous system development, and that's what we are very interested to know, at least with animal model. I think human study will come within 5 to 10 years, perhaps, or maybe longer. But I think we should be more proactive in learning about what CBD's impact in brain development. So I don't know how much you know about brain development, so I'll briefly introduce about mammalian brain development, and I already taught and how endogenous cannabinoid system has been shown to be involved in the brain, and the role of CB1 receptor, which we know most because there's a nice genetic transgenic animal mice available, and also genetic approach one can use, and of course lots of pharmacology, and beyond cannabinoid receptor 1, and pre or perinatal cannabis exposure can affect fetal brain endogenous cannabinoid system. And I'll tell you a bit about our data about the behavior outcome after the exposure during perinatal period. So this is proposed developmental roles. I mean, not every aspect has been studied in great detail, but it's shown that you have a lot more anandamide in the beginning of development, and then decline, and then go up a little bit. And 2-AG is increasing during development. And so you could see here the nanomolar versus picomole. So 2-AG and anandamide concentration in the brain are quite different. But it doesn't mean the more effective. So just kind of have a note here. And it's believed that it starts from the fertilization. Cannabinoid system has a role affecting the sperm. And then implantation, and as you see the neural tube, which is literally like a tube with single layer of cell in the beginning, and that anandamide and 2-AG begin to have impact on the precursor cell, the progenitor cells, for the neuronal differentiation, device proliferation and differentiation, and commitment to neuronal phase and migration and morphogenesis and axon guidance. I will show some of the evidence for the role of endogenous cannabinoid system on it. And Ismail, of course, has done a lot of this neurodevelopmental aspect. And it has also impact in adulthood for, of course, retrograde synaptic transmission aspect I mentioned about, and also for adult neurogenesis. So I hope I can play this movie for you. I want you to get appreciation about the development, how quickly things change. And so you could see, hopefully you could see the thickening of this part, the cortex. And so it's the events involved for these rapid changes involving first progenitor cells make more of itself, right? So there are more progenitor cells with symmetric proliferation because first you just have single layer, very, very simple tube, and become a complex brain. You need a lot of precursor cell. And then it transition to become a symmetrical division. So it will divide and generate a daughter cell that become post-mitotic neuron. So this so-called post-mitotic means they will no longer divide. And so this neuron born at this time are the neuron, majority of neuron in your brain. They are not going to get renewed. So this time, then there will be another phase of proliferation from intermediate progenitor cell, and during this time, they will all the proliferation stay in the ventricular zone next to the ventricle and subventricular zone, and then the daughter cell become post-mitotic neuron, move up toward the PO surface. And you could see here the transcriptional factors are very different at different stages. Okay, so just kind of and that sounds simple process lead to this complex structure, right, in all of you. And all this wrinkle can be zoomed in, and you could see very distinctive 6 cortical layer structure for majority of us, and that and this 6th layer has a purpose. Okay, so they are very, very organized into different structure and function and transcriptomic. So this data are acquired from so-called patch-seq. So meaning you patch to individual neuron, okay, at different cortical layer, and you acquire electrophysiological property, and then you label a neuron to look at morphology, and then suck out the cytosol to look at RNA-seq data. And amazingly, you could cluster them based on where they are. So the cortical layer really determine what kind of they will become, and the transcriptomic kind of profile them in a way, in an amazing way, I have to say. And so this structure, the layer structure, are very important because every layer neuron, they will project to different places to find their target. So here I simply put out from Allen Brain Atlas for the connection from somatosensory cortex at different layer, and you could see they go to a very distinct target zone. Okay, there's a very stereotype depending on which layer you are and then where you will go. And this connectivity is essential for how we behave. Okay, and this is fundamental for our neural circuit, and I just want you to appreciate the complexity, and you should try to have some fun with Allen Brain when you have time. They have a lot of information there. Okay, so, sorry, too loud. So starting from single layer, right, the cell move up and down depending on the phase of cell cycle, and they come down to divide and send out their daughter cell up and begin to form this organization of layer. So they have cell division and migration and differentiation. And all this happen in the womb, right? And this is a time that cannabis can have impact, and endogenous cannabinoid system does express here, right? They are present. So there's tons of literature, including Spice, where I cannot cover everything. I'm just going to highlight a couple. So endogenous cannabinoid has been shown to regulate the proliferation and mitosis in the of a neuroprogenitor cell in the cortical ventricular zone and subventricular zone here. So here is BRDU incorporation. BRDU incorporate into DNA when cell undergo S phase of cell cycle, synthesized phase of cell cycle. And you could see then you could do a birth dating and look at how many cells are in S phase. And you could see majority of them are in the ventricular zone and subventricular zone here in a wild type. And it is also true for CB1 receptor knockout mice, but there's a significant reduction. And interesting, FAR, which is a degradation enzyme for anandamide, when you take it away, you have more anandamide. And it has an opposite effect for the number of BRDU positive cells. So providing very, very strong supportive evidence for the role of endogenous cannabinoid system for regulating the progenitor cell proliferation. And Ki67 is an antibody recognized cell undergo mitosis, and here you see a reduction in CB1 receptor conditional knockout in the cortex. So these are very, very strong evidence suggesting the role of cannabinoid system in proliferation. And here is a work from my lab previously. And so once a neuron differentiate, they need to find their target. They send out exon to reach their target during development. And here, cortex will connect to the thalamus. Thalamus will connect to the cortex. And so here, you can see these two will meet up here and then guide each other to the final destination. And so CB1 receptor is labeled in green here, and D-lipase beta, which is 2-AG synthesizing enzyme, you could see they are present in this pathway here. So you could see this is a pathway coming in and going from thalamus to cortex. So D-lipase alpha is very prominent in the thalamocortical, and then CB1 is more on the other side. And so MEG-lipase alpha is the degradation enzyme, which seems to be present in both sides. So here's a zoom-in view. So you could see the green is for the thalamocortical axon, and that doesn't have CB1 receptor, but CB1 receptor have the DEG-lipase beta colocalized. DEG-lipase beta is colocalized with the thalamocortical axon, but not with CB1 here. And MEG-lipase is present in both thalamocortical and corticothalamic labeled by CB1. So I just want to showcase that these systems are present at the time when exons are finding partners. Okay, and in the CB1 knockout mice, if one look at this pathway here, the exon fasciculate into a particular thickness, and that is thicker in the CB1 knockout mice. And that can be seen also when TSC is being treated to the dam during gestation day 5.5, when embryonic day 5.5 to 17.5 with 3 milligram per kilogram. This is work from Thibaut Hackney's lab, and you could see there's an increase of thickness of this bundle. Okay, and that is CB1 dependent. Although CB1 itself is already very, very thick, and TSC treatment do not get increase. So just briefly, this is just to show you that cannabinoid system is acting on the proliferation and then also the target recognition. And next, I want to use the whisker barrel pathway to showcase the role of cannabinoid system on the fine level of aberration of exon. So here, I think many of you have worked with rodents, and you notice a whisker, and it has very, very nice representation in all the pathway, the brainstem, thalamus, and cortex. So all these patches represent to one particular whisker. So it makes it very convenient to look at its organization. And so we have found that DEG-lipase alpha, MEG-lipase beta alpha, they are all present in the barrel cortex, and BGLUT2 is a marker for some cortical axon. So it is in this area, and in the knockout mice, it does have whisker representation, but it has bigger gap in between barrel. And when one look at individual axon in detail, this is some cortical axon. It's really not fun experiment to do. These are painful experiments. You label single axon from thalamus and then look at its aberration. You see they can reach their target and aberrate extensively just within one whisker domain, basically in a wild type. And that is not like it happened just precise. It requires a lot of pruning, and it's believed to depend on synaptic plasticity, like long-term depression-like process to trim it down to be precise. So that it requires activity-dependent refinement. And that you could see in CB1 receptor knockout, they did not get to have the refinement. And it's not just happening to CB1 knockout, but also happening in dead glyphosate alpha knockout. So you could see this extensive ablation when endocannabinoid system get messed up. And so here is just to show you the synaptic plasticity from this pathway from the thalamus to cortex. Normally, you could induce a LTD formation, but that is CB1 receptor dependent. And it is not from the cortex, potentially from the thalamus. And this is what I just showed you. And so during this early development, when axons are uprising, if they get exposed to CB1 agonists like Wnt, this subcortical axon ablation are reduced, greatly reduced. And TSC seems to do similar job. It has this similar impact, so it maybe perhaps agonist here. AM2A1 is an antagonist, it increased ablation. So again, it's just different type of impact. So I have mentioned to you the popularity of cannabis, and that is estimated used daily by about 7% of pregnant women in U.S. That is pretty high. And so maternal cannabis use associated with increased risk in psychosis, depression, and anxiety disorder in offspring based on the human study. So we want to have better understanding of the lasting impact of this perinatal cannabis exposure. And so we need to understand how the brain development in human baby versus rodent babies. Okay, so and if one based on the landmark for the brain development, the newborn for human is corresponding to the postnatal day 10 of mouse brain development or rat brain development. So it's not birth to birth. So if we want to mimic consumption in human for the full pregnancy, one should continue throughout the pregnancy and then also the first 10 days after birth. And so that's what we set to do. But it is not easy to kind of model human consumption in mice. So as you know, human consumption is voluntary and often inhaled, like this tiny one here. But it's really not easy to get mice to smoke, okay? And they are reluctant to self-administer cannabinoids, and it success generally requires stress, and it should be a happy things to do instead of stressful. Humans don't take it because it's stress. So high efficacy, and so it really requires a lot of training. So most of the time was mimicked not even with THC or CBD and IP injection. So it's a lot of problem, and inhalation is getting popular, but inhalation has been shown to be stressful. Even in vehicle, you see a change of behavior, not by the THC itself. So here is for the play behavior is reduced play behavior with vehicle type of puffing, and very similar to TSC, which doesn't make any difference between vehicle and TSC here or open arm entry. So it's not ideal paradigm. And so edible, and Ken has shown beautiful edible way of doing it, but it has variable absorption, and during pregnancy it's harder. So we don't like inhalation paradigm. And so what it's a compromise. You have to compromise. It's not perfect. So we try to be very gentle. We use subcutaneous injection, and Gabriel can tell you all about the procedure. And so we use dose about 3 milligrams per kilogram to give plasma level and brain level similar to what seen in human and non-human primates, and try to model pharmacology instead of voluntary, because they are just not going to take it by itself. And so, but we need to keep the limitation in mind when we interpret. So, and the challenge, and we measure it, of course, to make sure we make what achieved in human or non-primate fetal brain. So we want to mimic human, of course, ideally, but we have no data to base on. There's no fetal brain in human that we could use. So the compromise is to use the plasma, maternal plasma label to try to achieve. But it's not a lot for the pregnant women, so we aim for the plasma label similar to non-pregnant users. and non-human primates. So this is what we are trying to go for. So this is what we decided on. And so first we tried to do daily injection to dams and wanted to have the exposure through dams, through mom. And so we treat the dam from gestation day 5 till 10 days after birth, because we think that postnatal day 10 of mouse pup brain is similar to human baby. And then we did mass spec measurement to measure the quantity of phytocannabinoids. So we measure one time point embryonic day 18.5 before birth and postnatal day 10. And we did 3 different kind of treatment: THC 3 milligram per kilogram, or CBD, as I showed the popularity of CBD, and we're really interested in what CBD would do, and the combination as well. There are a lot of medical marijuana is actually equal mix 1-to-1. And so we use CD1 mice because it's our breast strength. It's closer to human in terms of genetic diversity, and also they are good mom. Okay. So less impact on the maternal behavior and good number of pups. And so, and again, I have mentioned that, and then we sacrifice for mass spec measurement 2 hours after last injection. We mean Gabriel. and so basically, so here embryo, he took a whole cortex postnatal day 10. It's bigger brain, so we took prefrontal cortex, about 50 milligram, and then did mass spec with Heather and Claire. And so here you could see the content at embryonic day 18.5 after multiple days from mom. So they never get direct exposure. It comes from mouse to placenta. And so this is the THC content in the pups, in the fetal brain. And it's very similar to those non-primate measurement. And interestingly, the combination THC plus CBD, THC is the same amount, 3 milligram per kilogram, except here you have added 3 milligram per kilogram of CBD. The content of THC get to pup's brain is double, right? It's quite surprising to me, at least, but maybe not surprising to many of you know about the metabolism. And CBD, on the other hand, is similar label alone, but combination push it down. So it has a lot of differences, and without measurement, one we wouldn't know what's going on. And as many of you have been talking about endogenous cannabinoid system, what we found is that THC treatment through them reduce endogenous 2-HE concentration in the embryonic brain. But CBD seems maybe a little bit augmenting, but not significant. And here, this is anandamide label, which is also reduced by THC, and the combination also lead to reduction of anandamide. So clearly has an impact on endogenous cannabinoid system, and this is, we only measure one time point, and it will need more study in terms of how much it go on. And so next we try to mimic the postnatal phase here, thinking that it will be in a womb for human. So it should be continuous same level, because in human you'll be still in a womb. So we want to make sure of that. And so here the dam was treated for 10 days after birth, the same concentration. And you could see that here is a label of THC and CBD to mom's brain, and pup's brain THC is very similar. However, CBD doesn't go in very well. And so they actually get to the milk, but they just don't go to the pup's brain. And so they don't go get, apparently CBD is quite poorly absorbed by the pups. So to maintain the same concentration, we decided to do subcutaneous injection directly to the pups. And so we want to find a dosage that provide consistent label, and we found that 1 milligram per kilogram give us a label similar to what they get from the dam treatment. So this is 3 milligrams is too much, and so basically this is what we have done. So hopefully you are convinced that CBD and THC to them get substantial label to the embryonic brain, and combination give higher THC level and reduce CBD level. And both CBD and THC are concentrating in milk. I didn't have time to show you those data, and administrating THC to them result in appreciable and functionally important level, but not CBD. And so then we decided to give to pup directly with 1 milligram. And there's still a lot of work to do, and so we need to understand all this fetal, maternal, pup, and phyto versus endocannabinoid level and duration. How does this endogenous cannabinoid system changes continue through adulthood? Okay. And but this is again, so I'm going to show you some of the data has been published, and this was a time when we didn't know CBD doesn't get absorbed. So they were just inject to the dam throughout. Okay, so this CBD is likely to go to embryonic brain, but not postnatal brain. Just keep that in mind. And so first we were interested in the pharmaceutical pathway. As I showed you in the barrel study, we know pharmaceutical axon will be affected by endogenous cannabinoid system. So we want to see how does it look like in the prefrontal cortex. So MD thalamus to prefrontal cortex pathway is very, very important circuit for working memory and has been implicated in human for psychosis. And so that's why we focus on this. And so we use VGUT2 as a marker to look at the innervation of pharmaceutical axon in the medial prefrontal cortex. So this is coronal section. And here you could see VGUT2 seems to be increased in the male prefrontal cortex, but not by CBD or combination. So again, this is preliminary. It needs to be repeated to make sure. And this is consistent with many other people's work. Myeline binding protein is reduced significantly by THC and also CBD and the combination. And so, and this changes is actually quite region specific, because if we go to somatosensory cortex, it's a different story. So the impact could be depending on sex and depending on the region. And so Izaki, when he was in my lab, he did a panel of behavior assay for the babies. These are the baby grow up to adulthood. I mean, never get direct exposure to phytocannabinoids, only get it through the dams. And so he did a lot, and so they are, most of them are actually normal. And this is sucrose preference test, and he found CBD increased the preference for sugar here, sucrose, and in combination as well, but only in male, but not female. And for marble bearing, which is a good behavior assay often used to look at compulsive disorder, because they will be digging more marble and repetitive behavior. And you could see that female marble, that THC progeny, like it to dig more as well as CBD. And funny thing is that combination don't do that, and we don't know why. Here I want to focus more on the medial prefrontal cortex mediated behavior. And so this is a cute drawing from Maggie in Ken's lab. And so this is novel object recognition. It's a working memory test. And so basically you train the mice first to have two objects are similar, and then they get bored with that, and then you put one novel object and see how much preference toward the novel object. And so for the vehicle treated one, you could see they prefer the novel object, so the distinction index is increased in the second trial, which is novel object here. But THC progeny do not have this preference. So as I have mentioned to you, the cannabinoid, endogenous cannabinoid label is reduced in this THC progeny, right? So one idea is then enhance it. And one way to enhance it is to activate mGluR5 pathway by this PAN, positive allosteric modulator, CDPPB, to increase mGluR5 activity, thus increase 2-HG production. And so here, as you can see here, in the vehicle group, good novel object recognition, and THC don't. But if you add this CDPPB before then the test, then you reverse the behavior deficit. So this suggests that the impairment may come from changes in endogenous cannabinoid system. And good news is it's reversible. And the other complicated assay, which I put a drawing here, but I don't think I have time to go through it. But each box is 1 day, so you could see this is really a terrible behavior essay for people to do. It's a lot of training and a lot of habituation. But it's a very good one because basically you are testing their working memory. So, so during force training, you put the food pellet in one arm, and then you block the other arm, so the mice could only go to one arm and get the food. So, and then you ask it after 10 seconds, where should it go? It needs to go to another side to find the food. And so, if it go to the other side, it will get food, but if it go to the same side, it will not. So, 10 trials per day to train them. And the last day, you just try to see how many times they make correct choice versus wrong choice, and with more and more time delay. And so here you could see that vehicle learn very well, right? The increase of success rate every day, 10 trials per day, and then multiple days, 8 days of training. And they and TSC also learn, but they are not as good. They consistently perform less well. And the last day, you instead of just 10 seconds, you increase the duration of time, because the longer it separate the trial, the more chance to forget, right? Just like us. And, if we don't do things right away, we forget. And so you could see here the separation, the difference become bigger when the time, the delay get longer. And so TSC really seems to impair. And this is like 2 to 3 months after their last exposure and never got the right exposure. So this is really lasting impacts on perinatal TSC exposure. And this is prevented, in a way CB1 knockout mice, there's really no big difference, but it's need more work. The other behavior assay I want to mention is force swimming test. Many of you are probably quite familiar with it, and so it's a controversial behavior assay in terms of depressive life, but it's a very good assay to test the efficacy of antidepressant. And so here, as you know, they don't like to swim, and so we just want to see if the immobility time are different between vehicle and TSC. And I don't know why I have the gold bar here, but TSC conversion, okay. But anyway, just focus on the dot, the white bar here. And so the male and female, the immobility time, they are very similar across different groups. So there's really no difference in the baseline. Then we test their response to fluoxetine with the knowledge knowing that cannabinoid system and serotonin system do interact. Okay, and so we want to know the interaction between cannabinoid system and induction, and how phytocannabinoid system change that interaction. And so you could see, as expected in the male, the control group, they do reduce immobility time upon fluoxetine treatment, but TSC, progeny, the male, and CBD male, and do not respond to it. And even the combination, it seems like there are 2 groups of them. Okay. And that's male, and here's female. I apologize for using blue versus pink. That's very stereotype. But that's the easiest color for me to choose. Okay, so here again, the female, the control group does respond to it, but again, the progeny from TSC treated them and CBD treated them do not have that. Okay, and so here's putting together. When we put together, the combination does not seem to be effective. So it's interesting. But you could see here, I want to point out the 2 separation groups. So there are a group do not respond to fluoxetine and one group respond. Okay, so knowing potential cannabinoid system are altered and hyperfunction, so here we want to see how can we restore it. Okay, and so here is the fluoxetine response. You know, it decreases immobility time, and from previous work, and M251, the CB1 receptor antagonist, could block this. So we know there are potential interaction about cannabinoid system. So we want to test FAR inhibitor, URB 597, which is known to reduce immobility time, but it has this inverse U-shape pharmacology. So here is again just for swimming test, you could see if just apply 0.1 milligram per kilogram, there's no impact, but 0.3 reduce immobility time, just like fluoxetine, but when you go higher, it doesn't. So you want to choose a suboptimal dose to look at it, because with understanding, thinking that maybe just need a kick, a kind of a boost in a way. And so we use the dose that do not trigger effect by itself. And so you could see here, 0.1 milligram URB, all these have phosphol and fluoxetine versus the one with fluoxetine. So you could see here the fluoxetine reduced in vehicle group and now also reduced in all these others. So it really rescued the behavior phenotype for this. So a conclusion, enhancing anandamide signaling by FAR inhibition restore fluoxetine sensitivity, and enhancing seems to restore novel object recognition, and enhancing mGluR5 signaling also restore social interaction in rats, which is Olivier Manzani's group, which I have not time to show. And of course, there are many, many remaining questions. So I just list them out here, and I'm sure many of you probably will ask me those questions. So you know I'm thinking about it. Okay. And so with that, I want to thank you, and I want to show you beautiful campus we have. Again, I'm trying to recruit you. And beautiful Brown County very close by, and I'm very, very grateful for the temperature here. And Jack Gill, who donated the money to endow our Gill Center. And I also want to advertise this symposium that we are about to host September 14th, that you could join us either in person or through kind of a video link virtually. And as you can see, we have clustered the famous scientists, including some of you in the room here. So, okay, so basically, any question I'm happy to take.
Thank you, Dr. Lu. Now, I think. So we have 10 minutes for questions before the next talk. And again, I would like to invite everyone, especially the students, to raise their questions. No.
Hello, my name is Carola Peta. I'm from Greece, and thank you very much for your interesting talk. Actually, I would like to know if it was really interesting that in this developmental window, neurons become post-mitotic and have differences in formation of dendrites and axons. So I would really love to know if there is also abnormalities in the proliferation rate of astrocytes in this developmental.
Yes, so that's a question Gabriel is working on, and he could tell you more about it. We are very, very interested, and as you know, that astrocytes begin to proliferate around the time it gets birth, and it differentiates at postnatal time point, and that's like the third trimester corresponding to human. And Gabriel has some promising data, they do change, and so yeah, he could show you more.
More questions?
Hello, Professor. Thank you for the amazing lecture. Trying to join all the information about the lecture of Professor Kemaki, and have you saw something about the neural development in the hypothalamus? Because I was thinking how this effect could repercute on the peripheral metabolism.
So first, I'm not a hypothalamus expert, and hypothalamus is very complex. So I'm kind of afraid to go there. And metabolic studies are very, very difficult to do it well. And we didn't really see big change of body weight except the first 3 weeks for TSC exposed progeny, which I didn't really show. They started with lower body weight, but then they pick up afterwards. So that's pretty much all the information I can share to you. But I would love to get this paradigm accepted and everyone can work on it.
Hi, Professor Lu.
Yes.
Thanks for the amazing lecture.
Yeah.
I'm just thinking, how you see the contribution of microglia to the developmental effects of phytocannabinoid exposure, since that recent evidence showing that endocannabinoid tones controls sexual dimorphism of microglia in perinatal exposure, and also controls some behaviors like social behavior in adolescent and adult mice. So
yeah, that is a very, very good question. So we haven't really looked at perinatal aspect of microglia, but microglia are already there from the very beginning of, in the brain. As you know, it comes from blood, kind of yolk sac, right? So very, very beginning, they enter the embryonic brain at very, very beginning of brain development, and then they reside there. So they will be exposed to it completely, except we haven't looked at it. And that's one of the things that Gabriel is about to look. But anyway, I put everything to him. But for adolescents, cannabis exposure, however, Ken's lab has looked at it, microglia, and they see quite significant changes. And the other thing that we see is the MBP change, which often being attributed to oligodendrocyte, but there are a lot of recent work showing microglia's involvement for myelination. Okay, so I would be very interested to explore that angle, how the microglia change to affect myelination. As you know, myelination is so important for our function, and so I think that really need to be looked at. Yeah, synaptic pruning. So we did see changes in that. So microglia definitely can potentially play a role there. I mean, you see VGLUT2 changes, which is a synaptic marker, right? So, yes.
Thank you. Then after this, we might have time for one more question. So here and then there.
Thank you very much for the very interesting talk. My name is Iker, by the way, from Spain. So I was wondering, because there was a lot of data, and maybe you said it and I missed it, but did you, this effect of the THC that you see in the behavioral models, did you antagonize it in vivo, like administering together THC and maybe rimonabant or something, and see that the effect is not seen in the model?
No, we haven't done that, and that's so basically this is just first phase to see whether there's a lasting impact with this paradigm, and some of them we show that it doesn't happen in CB1 knockout. It's not pharmacology, but it's taking away CB1 knockout. So it does suggest some aspect is mediated by CB1 receptor, but there are so many things could be affected by THC, and CBD is even worse, right? We don't even know how many targets it has, and so it's kind of hard to exactly pinpoint which one. Again, we are trying to mimic human consumption, right? And the goal is to find therapy to reverse them, because all this baby will grow up in 10 to 20 years, and we hope to be able to find ways to help them.
Thank you.
Yeah.
Hello, Professor. Thank you very much. That was very interesting, very interesting. My name is Gunther van der Walt. I'm from Autónoma in Barcelona. And my question is, well, the interest comes from a very recent paper published on the recruitment of neuroprogenitor cells and the role of neurons and microglia in sending the signals for the progenitor cells to actually go to their intended target. So have you done any study, or do you have any idea of any study on the role of the endocannabinoid system or cannabinoids in general on the recruitment and localization of the progenitor cells to their intended target?
I think this should go to Ismail. Do you know?
So what we know about the progenitors is that once they become post-mitotic, the migration of the neuroblast is impaired. About the ectopic position of progenitors, we already haven't seen that. So usually progenitors are located in the VZ and SVZ, and so they will have different proliferation rates and probably different symmetric versus asymmetric divisions. But in terms of position, they shouldn't be modified unless they are modified before that.
we'll just extend a little bit because there's a lot of questions and we are actually fine with time if that's okay.
I'm fine as long as you're okay.
Hi, great talk. My name is Livia from Brazil. I was wondering if there's a shift in balancing between 2AG and NMN that could impact the coping response to stress, and then the FI inhibitor, if other inhibitors could also present the same results, and then we could see the change between this balance.
Yeah, I mean, that's a very, very good point. And we have not measured with mass spec, so I really don't have answer for you. But I think it's always a balance. And so, but here, if we are kind of knocking down both levels, then I think maybe bringing out a little bit, that's what is helping. And so the question is whether if we enhance 2AG signal by inhibiting MEK lipase alpha will also achieve the same effect. And we haven't done that experiment, but that's something we propose to do, and we are submitting a grant actually on that. I will talk to you more later.
I have a question. I'm not a student yet, though, but I will.
No more student.
So thanks for the data that you showed. So
yeah.
Usually before we were kind of assuming the hypothesis that prenatal cannabinoid exposure was impairing mainly CB1 receptor signaling.
Yeah.
And you're showing us more changes in endocannabinoid levels, right?
Yeah.
So my question goes towards the impact of cannabinoids on the epigenetics of the enzymes metabolizing the endocannabinoids. And I want to put a focus on how do you believe the importance in the progeny of the male, of the father and the mother? Because I believe, I mean, in terms of Jasmine Hart showed some impairment on
yeah.
TNC, and I believe, like, in terms of making social interpretations on, policy recommendations, like trying not to push, like, we are mimicking,
yeah.
we try to mimic prenatal cannabinoid exposure.
Yes.
But we are just looking on one side, and sometimes we are just putting all the guilty, or I mean, like, to me it's kind of real. I mean, I'm trying to avoid like being questioning from outside as scientists, what pregnant women do.
Yeah.
And why we're not seeing always, it's just like a question.
Yeah, and that's why we focus on therapy aspect of it, right? I mean, it's not to put anyone to blame, but we do need to have a solution for their progeny who later on have kind of differences or suffering, and so how can we help them? Yeah, and we have not looked at it, right? And so there are definitely a lot of studies showing that from sperms. Yeah, and it could be even microRNA in the sperms. And it could be even just stress. It doesn't have to be drug exposure to lead to lasting changes. And the one I know, actually, the father carry the bad microRNA and then lead to the daughters have problem. and then that's transgenerational, which is very, very interesting. And so it's not just women to blame. Yeah, but I think it's more the susceptibility of brain development, right, in the womb. And I think that's what I'm trying to emphasize here. And CBD, it may not be so, I guess, harmless in a way during pregnancy. It could be good for many other times, but I think just like anything, alcohol, we are not supposed to consume. And caffeine was the hardest one for me when I was pregnant.
So, hello, and thank you so much for sharing this amazing data with us. I was wondering, of course, we are talking about like wound neurodevelopment, but then you analyze the pups afterwards, right? Mm-hmm. And then you see the antidepressant effects, the lack of antidepressant effects of fluoxetine. So I was wondering if you also looking at other brain different regions, because you focus on the prefrontal cortex, but then we have the hippocampus and we have the other sides of adult neurogenesis. So what do you think is happening?
I think that a lot happened. We are systematic injection in a way, right? Subcutaneous, and it goes through the rest of the body as well. We just have to stay focused with small group of people, and that's why we're recruiting people to work with us. And of course, the paradigms we have is established in a way. If any of you feel this is a good paradigm, everyone can go for it. And so together as a team, we could figure out every aspect. Yeah, I mean, we just cannot do everything. Yes.
So we have a last question here, and then we move to the next one.
Sure.
Again, from the clinical perspective, what is the if you have any hypothesis to explain the difference between CBD and THC when it's taken by breastfeeding? Because THC is apparently is not detected, but THC yes. And we know that the fluxus pumps in the gut have a role with the CBD and THC different. Could it be explaining the absorption of the CBD? Because in this case, it would be a fantastic tool to treat mothers without harming the kids. You understand? And there is a huge polemic around this. And having this data could be really. But
I mean, again, we are using rodents, so we need to be careful about that. And I don't think we could take some baby and measure that. So this is, of course, a great news in terms of CBD reduced anxiety, post-mortem anxiety issue will be very helpful with CBD. And so, we were shocked to see, and that's why we check on the milk, and it's tons of them there. It just doesn't get absorbed by the pups. And whether this happened to a human baby, I cannot tell you. And so we think it's absorption aspect, but it could be also possible that CBD just somehow get metabolized very quickly. We don't really know for sure. Yeah.
Thank you, Dr. Lu. Thank you for the brilliant talk and also for answering all the questions. So now we would like to invite the next speaker, and unfortunately Manuel Guzman could not be here with us today, but then we'll have a very brilliant, promising young investigator from his group that will give us a nice talk on this topic, and that is Carlos Costas. Thank you for accepting this challenge. Yes, please. You can take this one.
Yeah? Okay. Yeah. Okay, thank you, Isma. As Samia said, my name is Carlos. I work with Manuel Guzman and Isma here in the School of Biology. And well, first of all, I want to apologize on behalf of Manuel. He has had some personal issues lately, and he couldn't come, and in fact positive for COVID as well. So it was really difficult for him. Today I just want to give an overview on the work we have been doing in the lab for the last 5 years or so, and I want to give an overview on the 3 candidates we have been working on. So I think Ken and Lou already introduced the cannabinoid system, the endocannabinoid system, so I don't really have to go deep into it. Just as a reminder, as you know, cannabis sativa, and particularly this very abundant compound, tetrahydrocannabinol or THC, they activate a specific G-protein-coupled receptors, type 1 or type 2 cannabinoid receptors mainly, also other ion channels and other molecules. But we focus on CB1 receptor. Of course, there is the endocannabinoids and the enzymes, but today I will focus mainly on THC. So we are interested in CB1 receptor because it's highly expressed in the central nervous system, both in the adults and, as we recently saw, in the developing brain. As you can see here in this mRNA hybridization assay, there are high levels of CB1 receptor mRNA in the striatum, in the cerebellum, hippocampus, amygdala, cortex, hypothalamus, and if you take another cut, in several other brain regions. So CB1 receptor participates in many functions precisely because of this widespread distribution, movement refining or coordination, memory and learning, fear and anxiety, food intake, and many others. But perhaps in the last years, cannabinoids have been studied very extensively because they induce neuroprotection in a wide array of pathological conditions. For instance, Alzheimer's disease or Parkinson's disease, Huntington's disease, multiple sclerosis. In fact, many of these studies have been conducted by people here in this group of seminars. Also hypoxia ischemia, I don't know, this is ALS, also some rare syndromes that go with intellectual disability. But the thing is that this is, of course, cannabinoids, but in most cases, CB1 receptor is involved, and we know little about the mechanisms of action of CB1 receptor, in particular in these diseases. We know that sometimes it's downregulated or sometimes upregulated, but at the molecular level, the precise action of CB1 receptor is not known. What we do know is that CB1 receptor is mainly a presynaptic receptor, as you can see here in this electron microscopy micrograph from Giovanni Marsicano's lab. And what the best characterized mechanism of CB1 receptor, I think everybody in the audience knows about it, but is the control of neurotransmitter release. It's one of the major neuromodulatory systems within the brain. So basically, just as a brief reminder, when a neuron fires, either an excitatory or an inhibitory one, then endocannabinoids are synthesized. We know much more about the excitatory production of endocannabinoids, but it's assumed that it also occurs in inhibitory terminals. This is dependent on calcium, and then these endocannabinoids traverse the synaptic cleft and activate presynaptically located CB1 receptor, which in turn blocks further neurotransmitter release. And this can be at the short term, leading to depolarization-induced suppression of excitation if the cell expressing CB1 receptor is excitatory, or inhibition if the cell is inhibitory, or maybe some other forms of plasticity that last more time, leading to long-term depression or potentiation, or even metaplasticity. But this is not the only thing CB1 receptor does in the central nervous system, and I think from my point of view, one of the most strong arguments that support this idea that CB1 receptor has much more roles, is that CB1 receptor is expressed in many other cell types, not only neurons, and not only excitatory and inhibitory neurons, also colinergic, serotoninergic, but also astrocytes, microglia, vascular endothelial cells, oligodendrocytes, neural progenitors, and even some others. So from the molecular point of view, we have a picture much more like this from much work from Ken Makis, in fact. We do know that CB1 receptor is mainly bound to GIO protein subtypes, and CB1 receptor activation by, for instance, THC leads to this heterotrimer dissociation. Then the GIO blocks adenyl cyclase. This downregulates cyclic AMP production and reduces protein kinase A activity, leading to different events, mostly gene transcription. But also CB1 receptor can activate tyrosine kinases, such as soluble ones like SRC or receptors, other kinases such as PI3 kinase, mTORC1, or MAP kinases, also small G proteins such as RhoA, which is important for migration in neurodevelopment, and also ceramide synthesis or calcium-dependent signaling. So some years ago, we started discussing that we believe that CB1 receptor action, and I think there's much evidence in the literature, is cell type dependent. That is, that some of these pathways are more prominent in a given cell type than in others, and also in the context where this cell type is encountered, the physiological versus pathological state, and so on. So the idea when I began my PhD was that maybe some cell-specific proteins, soluble proteins, that by physically engaging CB1 receptor could activate or deactivate some of these pathways. And this could lead to this cell type-specific CB1 receptor signaling. In fact, this has been very recently reviewed by Natasha Grimsey and Caitlin Noyagawa from the University of Auckland in New Zealand. And I think there is strong evidence suggesting that many proteins contribute to proper CB1 receptor signaling. But if you read this article carefully, there is always the biochemical interaction. But no one really goes into which cell type is expressing the interacting protein. So we thought that there was a space for our work when we began this. So we tried to identify cell-specific interacting proteins, and to do so, we did 2 high-throughput screenings. So to identify these proteins, we used the largest surface of the receptor, which is exposed to the cytosol, which is here framed in the red box. This is the carboxy terminal domain of the receptor. As you know, it has other potential interacting sites. The intracellular loop 3 is also very large, approximately the half of the carboxy terminal domain. And we even tried, but we weren't able to get any hits with this region. So the first approach was a GIST2 hybrid assay. So this is conducted in the beer yeast in Saccharomyces cerevisiae, and it's basically an approach that allows the detection of interacting proteins via a colorimetric assay. So what we do is we transform yeast with 2 plasmids, and one of them has your protein of interest, in our case a region of CB1, the carboxy terminal domain, which is fused by genetic engineering to the DNA binding domain of a transcription factor. The most commonly used one is the GAL4. And then you have another plasmid that encodes for a given protein. You can do a protein of interest. In our case, we wanted to identify new proteins, so it was a library containing more than 10 million different ones. And this is fused to the DNA, the transcription factor activating domain. So basically what you get is if the 2 proteins interact, in our case CB1 receptor and the unknown binding partner, then the transcription factor is reconstituted and transcription is activated. And what you activate first is a gene that reverts an auxotrophy, so you can select when yeast do not, when the media has not histidine in this case, and then a reported gene, the LAC set, which is able to hydrolyze XGAL, and it renders a blue compound. So basically what we did was to look for blue colonies. I think, well, this has been done for many years with bacteria and yeast. Probably some people here has done similar experiments. And we found this protein, which is BIP, binding immunoglobulin protein, which I will introduce later. And then we also did an affinity purification mass spectrometry experiment. In this experiment, what we did was we got a sheep brain, we homogenized this, and then this homogenate that contains brain proteins was introduced in a chromatography column that contained the carboxy terminal domain of CB1 receptor immobilized. Of course, we kept a column without it as the control. And then we, after washing and several steps, we just elute the binding proteins, and using mass spectrometry after trypsin digestion, we get a list of approximately 60 candidates. And among them, we did several cutoffs based on statistics and biological functions, and we got 2 proteins. The first one is CRVN, the main topic of the PhD of Alba Hermoso, who is here, so if you have any CRVN doubts, you can refer to her. And the other one was GAP43, which has been the topic of Irene Maroto, and we have extensive data on this protein, and I'm not going to show, so if you want to ask, she's also here. So BIP, this protein has the alternative names of GRP78 and HSPA5. In fact, we initially started calling it GRP78, and when I attended Congress, everyone was thinking that this was a GPCR because of GPR, from the first talk, so I had to change the name. So this protein is a chaperone, mainly present in the endoplasmic reticulum, where it regulates protein homeostasis. Basically, it's the major chaperone that controls proper protein folding in this organelle. And when the folding capacities of the cell are exceeded, it just triggers the unfolded protein response and autophagy to preserve the cell viability. And if it does solve the problem, then it leads to apoptosis. It also is a calcium buffer in this endoplasmic reticulum, and also it can act as a pro-survival receptor at the plasma membrane and under different contexts. And then the other two proteins, CRVN, this is a slightly smaller protein, 442 amino acids. And the interesting thing about this protein is that a mutation causes a mild form of intellectual disability in humans whose inheritance is autosomal recessive. And this protein is not very well studied, but what is known is that it is the substrate adapter of the CAL4 DDV1 E3 ubiquitin ligase complex, and thus the main function of this protein within the cell is to ubiquitinate target proteins, in most cases leading to protein degradation. And recently, not recently, 10 years ago, but this really triggered the research on this protein, it was discovered that this is the pharmacological target of thalidomide, which was a teratogenic drug 50 years ago that caused, in fact, severe malformations in approximately 10,000 children. Finally, GAP43, or growth axonal protein of 43 CannaLatan, is the smallest of the three proteins, 238 amino acids, and is mainly important for proper axon guidance, connectivity pattern, and neurotype growth during neurodevelopment. And in the adult brain, it's downregulated, but it's still present and in some brain regions, and it's involved mainly in synaptic plasticity and neurotransmitter release. So, of course, we had the three candidates, and now I will just go briefly on the questions we wanted to ask. Again, if you want, we have much more data that the data I'm showing today, but if you want to ask, we can discuss now or later. So, of course, the first question was to validate interactions. We got them in a high-throughput assay, and we really wanted to assess if they were true CB1 receptor binding proteins. So to do this, we do several technical approaches. I'm just going to show one per candidate protein, but of course we do every single one to all of them. The first one is fluorescence polarization. So what we do here is we recombinantly produce in E. coli the CB1 receptor CTD and our protein of interest, CAP43, CRVN, or BIP. And then we label the CB1 receptor CTD with a fluorophore. So in solution, when we excite this fluorophore, it emits light in all directions of space. So what we do in this experiment is we collect the emitted light in just one given angle, that is polarized fluorescence. And when the fluorophore is freely rotating in solution, that light is not really high. The basis is that if you get a protein to interact with your fluorophore-containing protein, in this case, then the rotation is slower, and then the polarized light you're getting starts to get higher. And if you fit this to a binding equation, you get information about the binding. Then the other type of experiment is a co-immunoprecipitation. I think this is very general and most of you know about. In this case, we have a cell lysate where CB1 receptor is present and presumably interacting proteins. And then you add an antibody against CB1 receptor, or in most cases an artificial tag. And then you add just a mix of protein A, G proteins, so you can increase the mass of these antigen epitope complexes, and then through centrifugation you just take your protein out from the mix. And finally what you do is Western blotting, and you look for the other protein when you, for instance, pull down CB1 receptor. And finally, we have also done proximity ligation assays. In this case, this is very similar to a conventional immunofluorescence, but the only difference is that the secondary antibody contains a DNA tie. And the thing is that if your candidate proteins are very close in space, these DNA ties can hybridize, then you can amplify by adding a polymerase, and you can detect these polymeration events with a fluorescent probe. So basically, if you get fluorescence, that means your proteins are very close, and this is consistent with an interaction. So this is the typical data we get. For instance, in the case of CRVN, you see this type of saturation curve, similar to a Michaelis-Menten equation, and you get the dissociation constant, which is in the low micromolar range. This is normal for a strong protein-protein interaction. For instance, in the case of BIP, here when we pull down CV1 receptor in HEC293 cells, then you pull down BIP, or a small region that we identified is the CV1 receptor binding site, and not when CV1 receptor is not present in the sample. And in the case of PLA, what we usually find is this type of red dots. This is in HEC cells that express GAP43 by transient overexpression, and they stably express CV1 receptor. And of course, we have all the controls. I did not put them here for the sake of simplicity. So the next question, we had already validated that they were CV1 receptor binding proteins. So the idea and what we wanted to see is that they do impact CV1 receptor signaling. So we did some functional assays in vitro to assess different aspects of CV1 receptor signaling, some of them similar to what Maki showed in his first talk. So as a general screening, we employ this technique, which is quite new. It's called dynamic mass redistribution. And in this approach, what we do is we plate the cells in a specific plate, sorry for the redundancy, which has a specific surface in the bottom of the plate. And this is resonant waveguide, I think is the technical term. But basically, this wave allows the crossing of the light in only one specific wavelength. So the basis is that you have a given cell type that expresses one receptor, and when you activate this receptor, then there's a change in the interaction of this inner bottom part of the cell that is in contact with the wave. There are some adhesion changes, cytoskeleton reorganization, proteins that get recruited to the vicinity of the receptor, and this changes the wavelengths of the light that is allowed to cross. So basically this is quantifiable, and you can get a curve that informs you about the global action of CV1 receptor signaling or whatever receptor you want to analyze. So we do this in HEC293 cell lines that stably express CV1 receptor, and we transiently transfect our given interacting proteins. And what we saw is that BIP completely inhibited the signal we see when we activate CV1 receptor with a selective agonist, which is the drug Win. Whereas when we overexpressed CRVN or GAP43, we saw only a partial inhibition, and in fact, the effect was slightly different in both cases. So the idea here is that, okay, all three proteins inhibit CV1 receptor function, but given that the curves are not really the same, and the acute effect is much higher, for instance, in the case of BIP, we believed that they impact different signaling pathways that we cannot assess by this technique, because this is a global signaling method, but we can, after this, dissect with other type of approaches. So we did several of these. In the case of BIP, this is I think the most visual one. Here what we did was we overexpressed BIP, and then we activated CV1 receptor with Win at 2 different time points. And then we did a proteomic array in which we analyzed the phosphorylation status of approximately 50 kinases within the cell. In this case, in most cases, phosphorylation means activation of these kinases. And as expected, CV1 receptor mainly activated kinases related to the PI3 kinase, mTORC1 pathway, and also the ERK pathway. But the interesting thing was that BIP overexpression completely blocked these two pathways. In the case of CRVN, what we did was to study the canonical pathway, which, as I said in the very first slide, was the blockade of cyclic IMP production. So for this, what we do is we treat the cells with forskolin, which is a drug that activates adenylyl cyclase and mobilizes cyclic IMP. And in this case, if we pre-activate the cells with a CB1 receptor agonist, then this cyclic IMP accumulation is reduced because adenylyl cyclase comes from a pre-inhibited state. So this is what we see in the control curve, as you can see, with increasing doses of CB1 receptor agonist, the drug CP, forskolin mobilizes cyclic IMP in a lower manner. But when we overexpress CRVN, this mobilization is reduced. So it seems like CRVN blocks this canonical GIO adenylyl cyclase pathway for CB1 receptor. And finally, for GAP43, well, mainly it's not really in HEC lines where we have got the most interesting data. It comes from electrophysiological studies, but I won't show. But basically what we have seen here is that it displaces G proteins from CB1 receptor, mainly GI2, GI3, and also GQ11. So basically, the conclusion was that they all are inhibitors of CB1 receptor, but they are selective inhibitors and they bias CB1 receptor action. So of course, the idea all around was that this protein shape CB1 receptor function, but they do in a cell type specific manner. And so far, I've just shown that they interact in vitro and they inhibit CB1 receptor in a heterologous system with overexpression, so we don't really have a cell type specificity there. And it's quite artificial, let's say. So we wanted to map these protein-protein interactions in the brain and specifically in neuronal cell types. So to do so, we took advantage that we already knew about proximity ligation assays. And the good thing about these assays is that you can do them in vivo. You can take a brain slice and you can, it's like an immunofluorescence assay, so you can do this. So we combined this with some mice we have in the lab, and we made brain slices of CB1 receptor conditional knockouts in which from a CB1 receptor fluxed background, we take out CB1 receptor using Cray-LoxP, and we take it from GABAergic cells or glutamatergic cells, mainly from forebrain, or every cell type using a ubiquitous Cre, or we don't take it at all and we keep the mice as controls. But we also had these mice where CB1 receptor contains a transcriptional stop cassette inserted in the open reading frame. So these mice are CB1 receptor knockout. They do not express CB1 receptor, but this stop cassette is fluxed. So we can take it out and we can rescue CB1 receptor expression levels, and this is similar to a wild-type mice. It's not overexpressed. And then, depending on the Cray we use, we can, of course, rescue it in GABAergic neurons, in glutamatergic neurons, in every neuron type, or just not rescue at all and get it as a control. So the idea here, I think most of you probably have deduced what we want to do, but just as an example, if we get, for instance, positive signal in a GABA CB1 receptor knockout, that indicates that the interaction is not happening in GABA cells because CB1 receptor is not there. And on the contrary, if we get a positive signal in, for instance, a GABA CB1 receptor rescue where CB1 receptor is only present at this cell type, then this interaction is happening in these cells. And of course, the data has to be consistent, and if you, this for instance wouldn't be, right? If you get this type of signals, then it's probably an artifact. So we did this for BIP. I don't know, I always have trouble by seeing the red dots, but I think you see, or at least I do. So as you see here, we have red dots here and red dots here, but not here and here. So that means that the interaction is happening when you have GABAergic CB1 receptor, because it's absent in the CB1 receptor knockout and in the GABA CB1 receptor knockout. And somehow expectedly, when you rescue CB1 receptor expression in every cell type, you get this type of dots. And when you rescue it exclusively in the GABA neuronal subtype, you get, of course, dots, and the most important thing is that you rescue completely the expression. So this indicates that this interaction is exclusive to these type of cells. And to be honest, we already had a hit because this protein had already been identified as associated to CB1 receptor in GABAergic hippocampal cells. In the case of GAB43, it was completely the opposite. We found that the signal was present when CB1 receptor was expressed in glutamatergic cells. Here, the signal is much lower. I don't think you can see. There are some red dots there. But anyway, here and here there is signal, but not in the CB1 receptor knockout or the GLU CB1 receptor knockout. And again, in the rescues, the data was consistent, and we rescued completely the expression of CB1 receptor GAB43 complexes when we have functional CB1 receptor in glutamatergic neurons. And for CRVN, the data is still ongoing because we have had many troubles with CRVN antibodies. They are not really good ones, but it seems like it's glutamatergic CB1 receptor. This is not coming from PLA data, but I will show you why in a few slides' time. So basically, we already knew that these interactions were cell type dependent. So the final question was if the data we were seeing in HEC cells would translate to these neuronal subtypes. So we wanted to know if they modulated CB1 receptor action in vivo, and we did functional assays in vivo. So here it was tricky because, of course, CB1 receptor is involved in a myriad of pathways. So where to look? And the thing is, we focus on the literature and previous knowledge, but of course, maybe some here in the audience now says, why didn't you look at this? Because it's kind of obvious, and of course, we are open to suggestions. So, for instance, in the case of BIP, we knew that CB1 receptor activation by a high dose of THC evokes anxious-like responses. And this is absent in GABA CB1 receptor knockout mice, so probably GABAergic CB1 receptor is necessary for this anxiety induction. And we knew that BIP inhibits CB1 receptor action very acutely in HEC cells, and we knew that the interaction was taking place in GABAergic cells. So it was kind of obvious to ask if BIP limits CB1 receptor evoked anxiety. So to do this, we employed the elevated plasmase. So you probably know about it. It's a cross-shaped device elevated from the floor, about 1 meter, 50 centimeters, and you have two corridors. One of them has walls, the other does not. And then if you have a very anxious mice, it wouldn't leave the enclosed corridor, whereas if you have a not very anxious mice, from time to time it will walk to the open corridors and take a look. So we did this in BIP wild type or BIP heterozygous mice. Unfortunately, the BIP knockout mice is not viable, and we wouldn't get access to the flox mice, which was our initial idea to do a GABAergic specific knockout. And we injected these mice with a high dose of THC, which should be anxiogenic. So we didn't get anxiety induction in the wild types, but what we saw is that this anxiety was very evident in the BIP heterozygous mice, which lack one allele of BIP. That means they have less BIP protein, which, as a reminder, it was a CB1 receptor inhibitor. So the idea here is you take out the brake, and CB1 receptor is able to fully activate this pathway. And I have to say that it's already known that this anxiety induction can be blocked by rapamycin, for instance, so mTORC1 dependent, and we know that BIP blocks CB1 receptor mTORC1 activation. So if you take out BIP, presumably this pathway exactly would be enhanced. So this was already published 1 year ago. And then we have GAB43. In this case, we turned our view to pathology, not normal physiology, let's say. In this case, we knew that CB1 receptor protects protects against drug-induced seizures, and this response is absent when you take out CB1 receptor from glutamatergic cells. And in this case, we knew that GAP43 inhibited CB1 receptor action in hippocampal glue cells. I have not shown, but Irene has done a lot of electrophysiology experiments precisely in hippocampal mossy cells, and we do know that it impacts CB1 receptors in active plasticity. So again, I think the question, it's obvious. The idea is, does GAP43 limit CB1 receptor evoked neuroprotection against seizures? So for this, we had to do much work, to be honest. First, we produced glue GAP43 knockout mice from a GAP43 flux mice using NEX1 CRE. And then we set up an experiment in which we inject vehicle or THC to these mice, and 15 minutes later, we inject a high dose of kynic acid. And this leads to convulsions and, in most cases, to death to these animals. So we monitor mice for 2 hours. We give them a score based on the Racine scale. And finally, we sacrifice the mice. So the key for this protocol was that in this setup, THC does not protect against kynic acid-induced seizure. The dose of kynic acid is so high, so basically in a wild-type mouse, and it doesn't even matter if they get vehicle or THC. So the idea here was again the same. If we take out GAP43, then CB1 receptor action should be facilitated, and maybe in a context where CB1 receptor does not protect, maybe we get CB1 receptor-mediated protection because its action is facilitated. And that was indeed the case. Of course, if anyone wants to see, I have the data from wild types, but here, as you see, THC does not protect, as I said. There is only a trend, but this protection is very evident in the glue GAP43 knockout mice. And finally, this is the last slide with data. In the case of CRVN, we took advantage that CRVN was already linked. Well, this is a mistake. This should be CRVN. In this case, we took the advantage of that CRVN is already linked to a disease. So basically, when there are mutations on CRVN in humans, this leads to enhanced CRVN-mediated auto-ubiquitination and degradation, and this in turn leads to cognitive deficits and intellectual disability with intelligent quotients around 70. So the idea is, if the mutations cause enhanced CRVN degradation, then maybe a cerebral knockout can mimic disease. So given that we knew that CRVN inhibited CB1 receptor action in vitro again, the idea, I want to just to showcase that the approach is very similar. In all cases, it's just the disease we are looking at. So the idea is maybe if we take out CRVN, and this is a model of a disease, if CRVN is a CB1 receptor inhibitor, maybe CB1 receptor overactivation underlies the disease. So in this case, first we generated CRVN knockout mice, and then we did a novel object recognition experiment, as Lou previously introduced in her talk. And basically here, what we assess is working memory by changing one of the objects. It's a long-term memory test. So here you can see the effect is very evident. CRVN knockout mice, they display a clear cognitive deficit. They cannot discriminate between the familiar and the unfamiliar object. And we also made conditional knockouts, and we found that the effect was mediated by CRVN molecules located on glutamatergic cells, but not in GABAergic knockouts. So basically, CRVN located, we think, is pyramidal neurons of the hippocampus, mediates this genetic deficiency-induced memory, well, amnesia. So the idea here was that maybe CB1 receptor in these glue neurons is overactivated. So what we did was to set up an experiment in which we took glue CRVN wild-type or knockout mice, and after the training session, when they have learned about the objects, we take a very low dose of rimonabant, a selective CB1 receptor antagonist, to see if blocking CB1 receptor can enhance memory recognition, or novel object recognition. So this was indeed the case. As you can see, we can totally revert the discrimination index impairment in the glue knockout mice when they are injected with rimonabant. So I will like to finish just, yeah, this was the correct slide. I don't know what happened. I would like to finish just by saying that CB1 receptor function is finely tuned by CB1 receptor interacting proteins, and for sure there are more. We have a list of 60. We have checked 3 of them. So we have, I don't know, 57 PhDs about to come. And these are the 3 ones that we have discovered. And then just saying that BIP inhibits global CB1 receptor function in GABAergic neurons and controls CB1 receptor evoked anxiety, whereas the other 2 proteins might be involved in pathology. GAB43 could contribute to proper seizure control by CB1 receptor. And for instance, I have to say that high levels of GAB43 have been found in epileptic patients. CRVN, on the contrary, might be a break for proper brain memory function by constraining CB1 receptor action. And I'd like to end here just by thanking all our collaborators from multiple universities and research centers, especially Irene and Alba, who have worked a lot in these proteins, all my group, and of course CannaLatan for having me here, and you for your attention. I speak a little Asian, so maybe Portuguese I can get, slow Portuguese. So yeah, I open to questions. Thank you very much.
Thank you, Carlos, for this excellent talk. And now we have time for questions.
Hello, Carlos. Thank you for this fantastic talk. I'm Israel, and I have a little question. You tried to observe this interaction with these 3 proteins in astrocytes, because we know that astrocytes express in more quantity this CB1 receptor, not only into the membrane, but membrane and in mitochondria.
Yeah, well, that's a good question. I did not show because I didn't have time, but we know, for instance, that the BIP action on CB1 receptor is mediated via GQ, so that immediately turned our view to astrocytes because, you know, this calcium-dependent signaling and so on. But I think the data on the proximity ligation assays, it really doesn't leave any room for, because you get total recovery, especially in the rescues. When you knock out CB1 receptor, well, you rescue total CB1 receptor expression, you get around 60% of the cells. Of course, there are astrocytes there, it's just a brain slice. And then when you do the GABA rescue, then it's also 60%. So I don't think there's room for astrocytes there. I think I forgot to mention, but the data of proximity ligation assay was in hippocampus. But we did that on striatum, on somatosensory cortex as well. And also, I think I showed CA1, and also we did the antigyrus. And it was always the same for the case of BIP. For the other 2, I do not know, to be honest. GAB43, I don't think it's much expressed on astrocytes. Maybe Irene can answer this better. And CRVN, I have checked in the past, but there isn't any paper. I remember I found one RNA-seq paper that said that it was expressed on astrocytes, but I don't think there is a single paper showing that at the protein level. So we will need to check.
Any more questions?
can hear.
Yeah, no, sure.
Very nice talk, and very close to my heart, all these protein interactions. Did you look at the distribution of CB1 in the BIP heterozygotes in terms of, is it trafficking affected?
Yeah, no, we didn't look, but we know, yeah, that's a good question, and we should have looked. What we know is that in heterologous system, CB1 receptor internalization is impaired. It blocks CB1 receptor internalization. And one line of research we haven't pursued, but I think it might be important for the future, is that I believe, and I think if you look in the literature, there's some evidence that CB1 receptor is bound to GQ. This has been shown in vivo in brain preparations. And I think CB1 receptor GQ activation might be important for CB1 receptor internalization in vivo. That's one idea for the future that I'd like to look.
Hi, very nice as usual. 2 questions. So why you choose the timing between THC and kainate acids for be only 15 minutes?
Yeah, well, I think that's the classical of, I don't know, maybe you know better, but I think Marcicano's earliest studies were 15 minutes. Yeah.
So will you wouldn't expect something different if you extend or short the timing?
Yeah, I mean, if you extend it too much, I don't think you are gonna get neuroprotection at all. I mean, if you go to, I don't know, 2 hours, something like that. Maybe, I don't know, maybe the peak is at 30 minutes and maybe you get more protection if you extend a little bit, but not so much, I would say. The thing is, well, one potential problem is like the first stages of the Racine scale are about the position of the mice, and and the thing is, if you get like a super hypolocomotive effect of THC, it can be tricky to differ whether it's THC or kainate.
Yes. And the next question is in the object recognition. Maybe I asked you before this. Do you also see something at short-term memory?
Yeah, we want to look at that. Yeah, we haven't checked, but
okay, so
yeah, of course, I mean, it makes sense because of your work to see if it's the one or
okay.
Yeah, we have to look definitely to that.
Thank you.
Thank you, Carlos. Congratulations on your data. Amazing, as usual. I was wondering if all those blind proteins, are they specific to CB1 receptor?
Yeah, another good question. What we do know is that they are not binding CB2 receptor. But for instance, I don't think it's necessarily bad that they bind other receptors. There are these data from, for instance, Wade Harper's lab, where they do human protein binary interactions. And GAP43, for instance, has been shown to bind sphingosine receptors, which I believe is a good thing because they are evolutionarily related to CB1 receptor. But again, this could be, I mean, the complexity can go even higher, and maybe if you get, for instance, GAP43 and sphingosine receptors, if you get more sphingosine receptors within a cell, maybe that's a way to displace GAP43 from CB1 receptor, and so on and so on. So yeah, what we always control is CB2. That's as far as we go. And well, we have checked, Irene, correct me if I'm wrong, I think adenosine A1 receptor and maybe some dopamine ones, but just as mere controls.
And just to add a little bit more complication on that, we know that we have populations of neurons in the hippocampus, for instance, that can release at the same time glutamate and GABA.
Yeah.
So in that case, and I know that we have results in the literature showing that they express CB1 receptors as well. So in that case, how?
Yeah. Yeah, I mean, I think we would need some more precise tools, because the CRILOX-P with NIX1-CRI, for instance, then you delete, mostly glutamatergic, but of course they can express more neurotransmitters than neurons you're employing. I think, for instance, GAP43, we have like a very complex viral system with 2 recombinases and so on and so on, and we are pretty sure they are mostly cells. I don't know about the heterogeneity of mostly cells. I think they are mostly glutamatergic. But yeah, in the other 2 cases, it could be different cell types. We haven't checked at all about neurotransmitter release in any of the proteins, which would be, of course, interesting if they really control CB1 receptor most well-characterized action. Yeah.
I have just one short question. Then
Yeah.
Do you intend to check, or have you checked whether there would be differences in terms of the subtypes of GABAergic neurons that could be modulated by the binding protein in CB1?
Yeah. Okay. Yeah, that's a good question. For BIP, yeah, if I had to take a wild guess, I would say CCK, because they express high levels of CB1 receptor in the hippocampus, but we haven't checked. One thing I was really confused when I had my data is in CRVN, I saw an RNA-seq paper of interneurons in hippocampus, and I don't remember correctly, but I think CRVN is one of the most expressed genes in CCK neurons. And that's weird because we think it's glutamatergic, but of course it could be like a change in excitatory inhibitory balance, and we, yeah, I mean, it's difficult because I think we lack maybe imaging tools that are much better to do so, but yeah, I think it would be important. Anyway, I think let's say the hardest part is done, because of course you can go deep and deep and deep at the molecular level, but I think the important thing is the output and the mice behavior, and of course in humans and so on. So yeah, I'd love to do that if we can.
Thank you. Yeah, one more question there.
Carlos, thank you very much. It's the first time I've seen many of these experimental approaches, and I love them. So I'd like to talk to you about that as well. But this is purely a curiosity question. So out of your list of 60 potential protein interactions, if you had the time and with what you know, what would be your VIPs that you think would be next on the list after these 3?
Yeah, well, of course, the first thing is you can compare with already published lists, so maybe one that overlaps between them. And also we have like private data from other labs that they have shared with us and us with them. So I would go for some of the proteins that are in common. But out of there is one protein, I mean, it's just a name, I will give you a name. There's one protein which is called LANCL2, which I think is a GEF, a GTPase exchanging factor, and I think I would go for that, just because of the function. But otherwise, I mean, it's, Irene and Alba can tell, it's very painstaking because you have to check the literature on every protein, and we had 60, and now, for instance, with Alba, we are doing another proteomics experiments, and we have 2,000, so you have to just read, and sometimes it's like a lottery. You just have feeling, a gut feeling, and you go for it. But yeah, we were very lucky that the three we selected were correct. Of course, that comes also for including controls and so on. But yeah, I mean, sometimes you're luck. You have luck.
Thank you.
Thank you.
So, any more questions? We still have time for. No? So, I would like to thank you again for this very nice talk. Okay, so we are ready for our next speaker of this section, and I'm very happy to introduce Dr. Carolina Echeverri. She comes from Uruguay, and she's associated researcher at the Department of Neurochemistry at Research Institute Clemente Stable Biological Research Institute, okay, in Montevideo, Uruguay. Thank you very much for accepting our invitation, and Dr. Carolina will speak in Spanish, but everyone is welcome in the end to ask questions in any language. Thank you very much.
Bueno, muchas gracias. Voy a hablar en español, voy a hablar lento para que se puedan entenderlo. Muchas gracias Ismael y Samia por la invitación y por dejarnos presentar los resultados que hemos obtenido en nuestro laboratorio en los últimos años. Bueno, entonces, como dice el título, les voy a hablar de las propiedades neuroprotectoras de extractos de cannabis y sus cannabinoides en estudios in vitro, son estudios en cultivos celulares. La charla la voy a organizar primero dando conceptos generales sobre la neuroprotección, sobre los fitocannabinoides como agentes neuroprotectores, para después pasar a mostrarles los resultados obtenidos en nuestro laboratorio sobre fitocannabinoides aislados purificados, la combinación de algunos de estos fitocannabinoides purificados, y después vamos a mostrarles los resultados obtenidos con extractos en base a extractos de cannabis. Particularmente vamos a mostrarles resultados de full spectrum de productos de uso médico y full spectrum de extractos realizados por nosotros en nuestro laboratorio, y terminaremos con las conclusiones. Bueno, entonces, cuando hablamos de neuroprotección nos estamos refiriendo al conjunto de medidas terapéuticas que van hacia limitar o retrasar lo que es la muerte neuronal. Este término se usó primero para el tratamiento de isquemias cerebrales, en la cual, a consecuencia de esta patología, se genera un núcleo isquémico llamado COR, donde la muerte neuronal ahí es irreversible, y donde no van a ir dirigidas las terapias neuroprotectoras porque las neuronas ahí están muy comprometidas. Sin embargo, alrededor de este núcleo hay una zona que se llama penumbra, donde el flujo sanguíneo no es tan reducido y la muerte neuronal sí puede ser prevenida, y es donde las terapias neuroprotectoras están dirigidas. Cuando hablamos de neuroprotección también lo asociamos con el tratamiento de enfermedades neurodegenerativas que, como ustedes saben, están caracterizadas por la muerte progresiva en diferentes regiones del cerebro, que según donde sea la región en donde estas neuronas mueren es que aparecen las diferentes enfermedades neurodegenerativas. Como ustedes saben, es una de las principales causas que lleva a la muerte en países desarrollados. Hay datos epidemiológicos que indican que hay un incremento significativo en los últimos años, y este aumento va a ir incrementando aún más. Esto demanda altos costos, tanto emocionales como económicos, tanto para el paciente como para el ambiente que lo rodea, y hasta el día de hoy no hay terapias disponibles que retrasen la muerte neuronal de estas enfermedades, sino que están todas destinadas a tratar los síntomas. El mayor problema que tenemos con las terapias neuroprotectoras en las enfermedades neurodegenerativas es el momento en donde se aplican estas terapias. Esta es una gráfica donde se ve la muerte de células cuando aparecen estas enfermedades neurodegenerativas, que es muy abrupta, y cuando el paciente aparecen los síntomas y va hacia las consultas, ya tiene aproximadamente un 60% de muertes celulares en las diferentes regiones según las enfermedades. Entonces, lo que las terapias neuroprotectoras tienen que ir asociadas junto con un diagnóstico precoz de estas enfermedades para que las terapias neuroprotectoras sean efectivas. En los últimos tiempos también se ha visto que los agentes neuroprotectores también pueden servir para tratar desórdenes neuropsiquiátricos. Si bien estos desórdenes históricamente han sido conceptualizados como disturbios neuroquímicos en estos desórdenes como esquizofrenia, desorden bipolar o la depresión, hay datos suficientes que muestran en pacientes post-mortem o en estudios de imágenes de cerebro que hay cambios morfológicos cambios morfológicos en el cerebro de estos pacientes, donde se ha visto que hay agrandamiento de ventrículos, reducción volumétrica y hay muerte de neuronas y de glías en regiones específicas como la región límbica y cortical en estos pacientes con este tipo de desórdenes. Además se ha visto que varios de los fármacos que se utilizan para tratar estos desórdenes han mostrado ser neuroprotectores en modelos de neuroprotección. Entonces el paradigma de que estas enfermedades simplemente los fármacos estaban dirigidos hacia equilibrar de alguna forma los disturbios neuroquímicos de las vías neuroquímicas que pasan en estos desórdenes está cambiando hacia que capaz estos fármacos lo que están haciendo es desencadenando ciertas señalizaciones neuroprotectoras que de alguna forma restablecen las vías neuroquímicas que están alteradas en estas enfermedades. ¿Y por qué? Entonces, ¿cuál es la razón de por qué los agentes neuroprotectores pueden ser útiles en enfermedades tan diversas? Y básicamente es porque los eventos bioquímicos que llevan a un mal funcionamiento celular y posteriormente a la muerte celular es básicamente los mismos. Y globalmente los podemos definir como que va a haber neuroinflamación, en donde va a haber activación de astrocitos y microglia, con liberación de sustancias como citoquinas o sustancias proinflamatorias. Va a haber estrés oxidativo. Ustedes saben que el cerebro es uno de los órganos que más sufre al estrés oxidativo. Tiene un alto contenido en ácidos grasos poliinsaturados, que son susceptibles a la oxidación. Tienen un alto consumo de oxígeno, las neuronas cerebrales, y eso hace que la homeostasis redox de las células, de las neuronas, sean muy sensibles a cualquier cambio intracelular frente ya sea un aumento de especies reactivas del oxígeno o un aumento en las defensas antioxidantes. Además, el cerebro tiene bajas defensas antioxidantes comparado con otros órganos. También hay difusión mitocondrial. La difusión mitocondrial va a llevar a un desbalance metabólico y también a un aumento de especies radicalarias. Y además de todo esto, otro evento importante en estos desbalances celulares es la excitotoxicidad con una desregulación de la homeostasis calcio. ¿Cuál es el inicio de por qué la célula empieza a funcionar mal de todos estos eventos? No se sabe. Lo que sí es que están todas relacionadas unas con otras y una lleva a lo otro. Entonces, cuando uno estudia, los investigadores hemos preclínicamente estudiado moléculas que de alguna forma mitiguen o limiten alguno de estos eventos para poder neuroproteger nuestras células, ya sea usando antioxidantes para regular el estrés oxidativo o la disfunción mitocondrial, ya sea antiinflamatorios para regular la inflamación o antiexcitotóxicos, si bien en algunos ensayos preclínicos han funcionado, cuando pasamos a la clínica, ninguno de estos fármacos han funcionado. Al día de hoy no existe ninguna droga que sea lo suficientemente efectiva para usar como neuroprotector en enfermedades neurológicas, ya sean crónicas o agudos. El problema principal es que la pregunta es hacia cuál de todos estos eventos bioquímicos que están sucediendo en la célula y llevan a un mal funcionamiento celular debemos dirigir nuestro fármaco. Hoy en día lo que está más aceptado que quizás para que el fármaco sea realmente neuroprotector debería necesariamente dirigirse hacia mitigar todos estos procesos que nombré al mismo tiempo. Y eso es lo que se llama estrategia polifarmacológica. O sea, agentes que tengan un amplio espectro y que sean capaces de actuar como antioxidantes, actuar como antiinflamatorios, como antioxitotóxicos y ya me olvidé. y como dije, antiexcitotóxico, antioxidante, antiinflamatorio y hacia la disfunción mitocondrial. o en su defecto usar combinaciones de agentes selectivos que juntos puedan cumplir esa estrategia polifarmacológica. Es aquí que entran en juego los productos naturales en general, en los cuales varios de investigadores han estudiado el efecto neuroprotector de varios productos. Los productos naturales son metabolitos secundarios y muchos de ellos cumplen con ese amplio espectro que les mencionaba anteriormente, sobre todo porque son buenos antioxidantes y buenos antiinflamatorios. La naturaleza tiene la particularidad de ofrecernos una gran diversidad estructural. Además, los productos naturales, los metabolitos secundarios, al ser sintetizados por en la planta por interacciones enzimáticas, tienen la capacidad de actuar, tienen ya blancos para actuar con sus proteínas, con lo cual lo hacen que sean candidatos efectivos para su neuroprotección. Nosotros específicamente en el laboratorio, la principal línea de investigación es la búsqueda de compuestos neuroprotectores de origen natural. Y trabajamos durante mucho tiempo en los flavonoides, particularmente en un flavonoide que es la quercetina, y por eso tenemos una gran batería de metodologías, ya sean in vitro, en cultivo o en vivo, para determinar y hacer screening de la capacidad neuroprotectora de diversos productos naturales. Y varias de las para buscar en dónde buscar estos productos naturales, los investigadores muchas veces nos basamos en los reportes que existen sobre el uso medicinal de estos compuestos como punto de partida hacia dónde ir. Y justamente una de las plantas que tiene una larga tradición en el uso medicinal es el cannabis sativa, que como ustedes saben, la cannabis sativa y el descubrimiento de sus compuestos activos dio lugar al conocimiento y a la detección del sistema endocannabinoide que tenemos. Entonces, el cannabis sativa, como todos sabemos, tienen más de 500 compuestos. Dentro de ellos tiene un grupo homogéneo, estructuralmente homogéneo, de meroterpenoides típicos del cannabis sativa, que fueron originalmente llamados como fitocannabinoides. La clasificación química está basada en que todos ellos derivan de un precursor común, el ácido de 21 carbonos, el ácido cannabigerólico, o su análogo de 19 carbonos, el ácido cannabigerobarínico. Como ustedes ven, todos los cannabinoides se producen en su forma acídica, que después, con el calor, se descomponen, pierden su grupo carboxila y forman sus formas neutras. Igualmente, hoy en día se han descubierto que hay varios compuestos, varios productos naturales que son capaces de actuar sobre el sistema endocannabinoide. Entonces, hoy en día la definición de fitocannabinoide, más que química, está definido como cualquier producto derivado de las plantas que son capaces de actuar directamente con el sistema endocannabinoide. ¿Cuáles son las razones de por qué los fitocannabinoides pensamos que son putativos agentes neuroprotectores? Yo les voy a nombrar 3 características que en realidad ya fueron disertadas durante toda la mañana. La primera de ellas es justamente su acción de amplio espectro. Como ya fue mencionado, los cannabinoides son capaces de actuar en el sistema endocannabinoide, pero también en otros blancos que no están presentes en el sistema endocannabinoide, lo que los hace ser agentes de amplio espectro. Esta, por ejemplo, es una figura de una revisión que publicamos recientemente, donde mostramos los mecanismos posibles de acción de 2 fitocannabinoides no psicotomiméticos, que es el cannabidiol y el cannabigerol, cómo tiene diferentes blancos de acción, ya sea dentro del sistema endocannabinoide como fuera del sistema endocannabinoide. Si bien CBD y CBG particularmente tienen baja afinidad por el CB1 y por CB2, ellos son capaces de actuar indirectamente en estos receptores a través de su modificación del tono de los endocannabinoides, actuando ya sea a través de las enzimas que degradan estos endocannabinoides o también enzimas que transportan estos endocannabinoides. Dentro del sistema no endocannabinoide también hay varios blancos, como por ejemplo el receptor serotoninérgico 1A, el transportador de adenosina, el receptor de adenosina, los receptores vaniloides y particularmente el PPAR nuclear, que es importante en la actividad neuroinflamatoria, y la actividad antioxidante también está dada por su acción directa en los radicales libres o su acción sobre factores transcripcionales que están relacionadas con la expresión de enzimas antioxidantes. Si bien nosotros hablamos de target o blancos dentro del sistema y fuera del sistema, hay varios investigadores que lo nombran como el sistema endocannabinoide expandido o endocannabinoidoma. La segunda característica que los hace ser putativos neuroprotectores es la localización y distribución de dónde se encuentran los blancos, sobre todo el sistema endocannabinoide. El receptor CB1, como ustedes saben, es el receptor metabolómico que está mayor expresado en el cerebro y se expresa en regiones donde en las enfermedades neurodegenerativas estas neuronas mueren, como en los núcleos basales, la corteza cerebral, el cerebelo, todas regiones asociadas con las enfermedades neurodegenerativas. Además, como fue mencionado, además de estar presentes en las neuronas, estos receptores están presentes en células del sistema nervioso como la glía, el astrocito y la microglía. Si bien el CB1 el CB1 está predominantemente expresado a nivel de la presinapsis, el CB2 está expresado principalmente a nivel de la postsinapsis y eso hace que sea un amplio espectro, además de tener el amplio espectro de acción puntualmente en las células, están en varias células y en regiones donde son necesarios. Además de estar, como fue mencionado, en la membrana plasmática, además está en organelos citoplasmáticos tan importantes como la mitocondria, que la disfunción mitocondrial dijimos que es un importante evento dentro de esa disrupción celular que ocurre. Si bien se sabe que la acción, sobre todo el THC, de agonistas del CB1 mitocondrial, lo que hacen es disminuir la respiración celular y disminuyen el AMP cíclico, no hay una relación todavía descrita exacta sobre el papel de este receptor en la mitocondria y la neuroprotección. Y por último, la tercera característica es que hay varios estudios que muestran que varios componentes del sistema endocannabinoide, ya sean receptores, ya sean los endocannabinoides o enzimas de síntesis o de degradación, están desreguladas en varias de las patologías que ya les mostré al principio. Todo esto hace que los fitocannabinoides tengan un alto potencial como nuevos agentes terapéuticos para tratar diferentes desórdenes del sistema nervioso central. Si bien ya he descrito la actividad neuroprotectora particularmente del THC y del CBD, todavía faltan muchos otros fitocannabinoides, como les mostré, que presentan el cannabis por descubrir sus efectos neuroprotectores y mucho más falta sobre los mecanismos de acción que están asociados al THC, al CBD y los otros fitocannabinoides que faltan evaluar su capacidad neuroprotectora. Esta es una revisión que hizo Stone en el 2020, en donde hace una revisión de la mayoría de los fitocannabinoides que están estudiados su potencial neuroprotector. Se ha estudiado en diferentes patologías, en donde algunas de ellas se han asociado con algún mecanismo de acción, pero como ustedes ven, falta mucho por descubrir y por descifrar, y lo que hoy en día carece también es de efectos comparativos, donde en un modelo se prueben varios fitocannabinoides para poder comparar su potencia o su eficacia. Es por eso que se requieren más estudios sobre el potencial neuroprotector de todos los fitocannabinoides que están por estudiarse y de las posibles combinaciones que puedan haber entre ellos para que puedan ser neuroprotectores más efectivos. Es por eso que nosotros, como les comenté, ya basados en esta literatura y que en nuestro laboratorio teníamos puesto a punto una amplia gama de metodologías para estudiar el efecto neuroprotector, es que nos propusimos primero hacer un estudio comparativo de diferentes fitocannabinoides aislados. Para eso lo que hicimos fue mediante una colaboración con Phytoplan Research. Ellos nos donaron los fitocannabinoides ya aislados y purificados. Empezamos a hacer el estudio comparativo con CBD, con CBG y con sus respectivos ácidos. Si bien en este momento la capacidad neuroprotectora de CBD ya estaba descrita, la capacidad neuroprotectora de CBG había muy poco y no existía bibliografía acerca de qué pasaba con sus respectivos ácidos. Entonces nos propusimos primero hacer en un modelo in vitro el estudio de la capacidad neuroprotectora de estos 4 fitocannabinoides frente a 2 estímulos oxidativos. El modelo que usamos fue cultivos primarios de neuronas granulares de cerebelo. Estos cultivos son muy utilizados para hacer estudios de screening, tanto de toxicidad como de protección de diferentes productos, ya que es un cultivo muy sencillo. Las neuronas granulares del cerebelo, al diferenciarse postnatalmente, se disecan luego del nacimiento a los 6 y 8 días, y eso hace que la disección y la obtención de estos cultivos primarios sea muy sencillo. Además se obtienen una cantidad muy grande de células, con lo cual en una misma disección se pueden probar diferentes moléculas y hacer justamente estudios comparativos y de screening para poder comparar su capacidad neuroprotectora. Además, como les mencioné anteriormente, el cerebelo es una de las regiones que está altamente expresado el sistema endocannabinoide. Como ustedes ven, acá se muestra el CB1 expresado en las neuronas, en los granos, y el CB2 también se encuentra en neuronas, pero principalmente en los astrocitos que tenemos estos cultivos. Estos cultivos, si bien cuando uno los realiza, el 90% de las células del cultivo son neuronas granulares, hay un porcentaje de astrocitos, un 5%, que quedan siempre cultivados debajo de nuestras neuronas. Entonces, el paradigma experimental que utilizamos fue, obtuvimos los cultivos celulares, sembramos, a los 6 días estas neuronas ya están diferenciadas, ya se conectan unas con otras a través de sus neuritas, hay sinapsis, y ahí empezamos a hacer nuestros pretratamientos con los 4 fitocannabinoides que les dije. Hacemos 2 tipos de pretratamiento, uno durante 24 horas y otro más corto de 1 hora. Estos pretratamientos lo elegimos en base a nuestro conocimiento que teníamos con los otros productos naturales, en donde hay ciertos productos que necesitan estar 24 horas para la inducción y síntesis de enzimas antioxidantes y antiinflamatorias, por ejemplo. En ese momento, entonces, después lo que hacemos es someterlas a las toxinas, que como les expliqué, utilizamos 2 tipos de toxina, la rotenona y el peróxido de hidrógeno. El peróxido de hidrógeno, como ustedes saben, es una especie reactiva del oxígeno que por él mismo no es muy tóxica, pero una vez que se forma rápidamente puede formar el radical hidroxilo, que sí es altamente tóxica, y lo que hace el hidroxilo es oxidar, dañar todas las macromoléculas, lo que se encuentra al lado en realidad, porque es altamente reactivo, ya sea DNA, lípidos y proteínas, y esto lleva a un desbalance, a una disrupción de toda la señalización redox necesaria para que una célula funcione, y lleva a la muerte neuronal. La rotenona es una toxina que es inhibidora del complejo 1 mitocondrial. En realidad fue un pesticida que se dejó de usar y se le asoció con el aumento de la prevalencia de enfermedad de Parkinson. Entonces, la rotenona, al inhibir el complejo 1 mitocondrial, produce una disminución de ATP, un desbalance metabólico de la célula, con un aumento de especies radicalarias que terminan también haciendo una desregulación de la homeostasis del calcio. Esas dos toxinas entonces las dejamos durante 24 horas y a las 24 horas evaluamos la viabilidad celular por dos metodologías. Una bioquímica, que es el método del MTT, que lo que mide es actividad mitocondrial, y después hacemos inmunocitoquímica. Primero les voy a mostrar los resultados que obtuvimos en cuanto a la neurotoxicidad por sí de estos fitocannabinoides. Acá tenemos el pretratamiento de 1 hora, en realidad dice 24 porque como los dejamos con la toxina 24 horas más, dejamos el mismo tiempo dejamos a nuestro fitocannabinoide. Tenemos el otro paradigma que es las 48 horas. Como ven, el CBD es un poco más tóxico que el CBG en los dos paradigmas, y los ácidos son un poco menos tóxicos que sus respectivas formas neutras, porque como ven, algunos en 20 micromolar recién empiezan a mostrar toxicidad. Este es el resultado que obtenemos frente al peróxido, el pretratamiento de 24 horas y después le agregamos el peróxido. En estas gráficas estamos representando la viabilidad celular como porcentaje de muerte. Acá serían las células controles y este que vemos al principio es solo con el peróxido, que como ven produce una muerte de aproximadamente un 60%, y el pretratamiento de los diferentes fitocannabinoides, que como pueden ver, ninguno de estos compuestos es capaz de prevenir el daño producido al peróxido de hidrógeno cuando es preincubado durante 24 horas. Cuando hacemos el otro pretratamiento, que era 1 hora y posteriormente le agregamos el peróxido de hidrógeno, aquí sí el CBD y el CBG muestran una protección significativa frente a los cultivos tratados solo con peróxido, mientras que sus formas ácidas, ninguno de ellos muestra acción neuroprotectora. En esta figura lo que vemos son no se ve mucho, vemos los controles, en donde están marcadas con hedge y en verde está la tubulina, en donde vemos el severo daño que produce el peróxido de hidrógeno, que si bien que el CBD y el CBG es capaz de prevenir, no totalmente, pero sí el daño severo que se ve con el peróxido cuando se preincuban durante 1 hora. Cuando se preincubaban durante 24 horas no había efecto. Posteriormente pasamos a la otra neurotoxina, que es la rotenona, que como pueden ver, la preincubación de 24 horas, tanto para el CBD como el CBG, son capaces de prevenir esta muerte neuronal de forma significativa, y en cambio ninguno de los ácidos es capaz de prevenir la muerte neuronal. Acá en realidad el CBDA a altas concentraciones muestra una prevención de esta muerte neuronal. Nosotros pensamos, y lo estamos identificando, que en realidad lo que está pasando es que a estas altas concentraciones y una incubación durante 24 horas, el ácido del cannabidiol se está descarboxilando, y que acá en realidad lo que estamos viendo es el efecto neuroprotector del CBD por sí mismo y no del ácido del cannabidiol. El pretratamiento, después pasamos al pretratamiento con rotenona durante 1 hora, y como pueden ver, tanto CBD como CBG son capaces de proteger la pérdida neuronal causada por la rotenona. Sin embargo, ninguno de los ácidos es capaz de prevenir esta muerte. Acá en estas figuras vemos exactamente lo mismo. Lo que vemos es cómo la rotenona produce un daño, pero no es tan severo como el que produce el peróxido. Y ustedes me van a decir por qué no cambié las concentraciones del peróxido para producir un efecto no tan severo y darle más oportunidad a que nuestros productos puedan proteger, pero el peróxido de hidrógeno tiene una característica que es o no protege, o no mata, o mata. Hay una concentración, y por eso los estudios que les voy a mostrar a continuación son básicamente en el paradigma de la rotenona. Continuamos porque la rotenona tiene un daño no tan severo y le da oportunidad a que puedan mostrar neuroprotección los diferentes fitocannabinoides. Y como ven, el pretratamiento con CBD o con CBG no son iguales a los controles, pero sí muestran una protección más efectiva que contra el peróxido de hidrógeno. Entonces, después vimos, como les dije, nos quedamos con el ensayo de la rotenona y pasamos a estudiar qué mecanismos de acción podrían estar asociados con este efecto neuroprotector. Y pasamos a estudiar los que ya estaban descritos en la literatura para el CBD y el THC principalmente. Lo que hicimos fue usar antagonistas del receptor CB1, el AM251. Como pueden ver, acá está la protección del CBD, y cuando es concubada con el antagonista, esta no bloquea la acción neuroprotectora del CBD. Lo mismo pasa con el CBG, lo mismo pasa cuando usamos un antagonista del CB2, que no revierte la neuroprotección ni por el CBD ni por el CBG. Lo mismo pasa cuando usamos antagonistas de PPARs, ninguno, ni para CBD ni para CBG, es capaz de revertir la neuroprotección observada por CBD o por CBG. Y cuando usamos el antagonista del receptor serotonina 1A, vemos que hay un cambio en la neuroprotección solo por CBG. Esto es contrario a lo que estaba en ese momento descrito para la literatura, en donde en realidad se ha mostrado que el CBD es antagonista del 5-HT1A y que por ahí podía ir su acción neuroprotectora, y el CBG en realidad está reportado como antagonista del receptor 5-HT1A, pero el único cambio que nosotros observamos en este modelo experimental fue ese. Entonces, basados en que estos compuestos, CBD y CBG, estaban produciendo neuroprotección y esa neuroprotección en principio no estaba dada por su acción ni sobre CB1 ni sobre CB2, pensamos, ¿por qué no hacemos combinaciones de fitocannabinoides puros con alguno que sí tengan acción sobre CB1 y CB2 para poder de alguna forma potenciar? Entonces, lo que propusimos fue hacer estudios de combinaciones de CBD y de CBG con THC y tratar de buscar diferentes relaciones para ver si de alguna forma podíamos potenciar ese efecto neuroprotector, porque como ustedes vieron, nunca fue un 100% y ni un 80%. Entonces, es un proyecto en el que estamos trabajando, que además de buscar las dosis, las relaciones entre el CBD y CBG con el THC, está propuesto, que no se los voy a mostrar porque todavía no lo hicimos, hacer estudios de biodisponibilidad mediante microdiálisis para ver también estas interacciones, cómo afecta la biodisponibilidad tanto del THC como el CBD, como fue mostrado anteriormente, ya que se metabolizan por básicamente las mismas enzimas. Entonces, seguramente esté variando la concentración en el cerebro, con lo cual su acción neuroprotectora también va a estar modificada. Entonces, lo primero que hicimos antes de ver la unión entre estos cannabinoides fue estudiar la neuroprotección por THC, que no estaba hecho. Como en esta gráfica de acá, lo que vemos, el paradigma experimental es, como les dije, con rotenona, y nos centramos para poder seguir estudiando las interacciones entre los diferentes compuestos en el paradigma solo de 1 hora, que en realidad es el que muestra mayor neuroprotección comparado con el de 24 horas. Como ustedes ven, la neurotoxicidad del THC es incluso un poco menor que la que observamos para el CBD, que el CBD ya empezaba en 10 a hacerse neurotóxico, y vemos que a concentraciones similares a las que muestran CBD y CBG, el THC también es neuroprotector y también es capaz de prevenir la muerte causada por la rotenona. Entonces empezamos a hacer las combinaciones con CBD o con CBG, a concentraciones en las cuales ellos por separados nos protegían. Hicimos relaciones de 1-1, de 1-2, de 2-1, de 1-1 para concentraciones más altas, y ninguna de las concentraciones que utilizamos, de las relaciones que usamos, potenciaba o adicionaba el efecto neuroprotector que mostraban los compuestos individuales por sí mismos. Lo mismo nos pasó con el CBG. Ninguna de las combinaciones que que realizamos muestran una potenciación o una adición de la capacidad neuroprotectora que mostraban los compuestos por separado. Esto simplemente que no lo había mostrado, que antes también habíamos probado si el CBD y el CBG ellos mismos se sinergizaban y tampoco mostraban ningún efecto aditivo. En base a que, entonces lo que quisimos es seguir estudiando sobre la interacción no solo entre los fitocannabinoides presentes en la planta, sino sobre los otros compuestos presentes en el cannabis, como son los terpenos y los flavonoides, una hipótesis que Russo nombra como entourage botánico, donde él propone que el efecto de la planta, del consumo de la planta entera va a tener efectos más beneficiosos que el consumo de un compuesto individual. Pero hasta el día de hoy no hay evidencia científica que muestra que haya realmente una interacción ni entre los fitocannabinoides ni entre los fitocannabinoides y los otros compuestos presentes en la planta del cannabis. Entonces, para eso estudiamos dos tipos de extractos basados en cannabis. Los dos full spectrum, uno es un full spectrum, pero es un producto médico que está comercializado, que se comercializa en nuestro país, y el otro full spectrum que probamos son extractos etanólicos que realizamos en nuestro laboratorio. Entonces, el producto médico que utilizamos para estudiar la capacidad neuroprotectora es el ipirfactán, que fue el primer producto que se registró en el Uruguay por el Ministerio de Salud Pública. Está registrado solo para el tratamiento de epilepsia refractaria en adolescentes y en niños. Este producto está de las dos formas, tiene un 2% de CBD o un 5% de CBD. Tiene altas concentraciones de CBD comparados con otros con los otros fitocannabinoides. Por ejemplo, contiene menos del 0,2% de THC. Lo primero que hicimos fue verificar en nuestro laboratorio analíticamente las concentraciones que mostraban tener este producto. Como ustedes ven, y esto es un cromatograma de un HPLC con arreglo de iodo a 210, que es la longitud de onda donde absorben los cannabinoides. En verde vemos el pico del estándar del CBD, y en rojo es el perfil de cannabinoides del ipirfactán, que como ustedes ven tiene principalmente CBD y después tiene trazas o cantidades menores de otros cannabinoides. Este, por ejemplo, es el CBDA y este es el THC. Entonces, lo que hicimos fue, en base a este análisis, a esta cuantificación del CBD, en todas las dosis que probamos, este producto no fue tóxico, con lo cual el espectro de neuroprotección que observamos con este producto es mayor, porque hasta las dosis más altas es neuroprotector. En estas fotografías lo que vemos es lo mismo, nada más que acá empezamos a marcar con GFAP los pequeños astrocitos, los pocos astrocitos que hay en el cultivo. Acá vemos otra vez la rotenona, como mata severamente, como el la ipirfactán es capaz de prevenir la muerte observada por la rotenona. Acá lo que hicimos fue cuantificar el área neurítica como una forma de mostrar la neuroprotección. Como ven, con la rotenona hay una disminución significativa de esta área neurítica, y el tratamiento con epifractán es capaz de prevenir este daño en las neuritas del cultivo. Esto es para que se acuerden que si comparamos este efecto del epifractán con el efecto del CBD, en donde se volvía tóxico a los 15 micromolar, y esto hacía que la ventana terapéutica de dosis sea menor, dado que después se vuelve tóxico, dijimos, hay un efecto diferencial entonces entre el producto y el CBD aislado y purificado. ¿Será esto debido a los otros productos que, si bien los fitocannabinoides estaban están en bajas concentraciones, pero también tenemos flavonoides, también tenemos terpenos. Entonces, lo que hicimos fue, en ese momento salió al mercado otro producto que es Salex, que es un producto farmacéutico, que es un purificado de CBD de la misma empresa, que como ustedes ven, acá tenemos el perfil de cannabinoides del CBD estándar, el epifractán, que ya lo habíamos visto, y el Salex, que como ven es un estándar básicamente, y lo que hicimos fue estudiarle la capacidad neuroprotectora de este compuesto para poder compararla con el epifractán, y como ustedes ven, tiene exactamente el mismo perfil de neurotoxicidad y de neuroprotección que el epifractán. Entonces, no son los otros compuestos presentes en el epifractán que le están dando esa propiedad de no toxicidad. Entonces, lo único que nos quedaba es el vehículo de formulación. Los dos productos están formados con el mismo vehículo de formulación, que es el MCT, es un triglicérido de cadena media, que es un vehículo que es utilizado para vehiculizar drogas lipídicas, y le evaluamos a ver si el MCT por sí tenía un efecto neuroprotector, y no tiene el efecto neuroprotector. O sea, lo que nos está haciendo el MCT es de alguna forma dándole la biodisponibilidad al CBD para que no lo presente todo de golpe y sea tóxico para la célula. Lo está liberando de manera que no es tóxico y permite una mayor ventana terapéutica. Por último, fuimos a los extractos experimentales realizados en nuestro laboratorio. Esto es una tesina de grado, que en realidad lo que estamos haciendo es evaluar el efecto neuroprotector de dos variedades, alfa y beta, que son las primeras variedades que fueron comercializadas en las farmacias de nuestro país. Estas flores fueron donadas por el IRCA, que es el Instituto de Regulación y Control del Cannabis de nuestro país. Y lo que primero que hicimos fue en realidad realizar diferentes extractos con diferentes concentraciones de etanol, diferentes tiempos de maceración, y nos quedamos con aquel extracto que en polifenoles totales rendía más. Ese extracto fue el extracto de etanol agua, realizado al 70-30%, que como ustedes ven, el perfil cromatográfico de este extracto hecho al 70-30% es el que está en negro, que como ven tienen un alto contenido en CBDA, un contenido menor en CBD y un contenido, la relación entre CBD y THC de estas flores es 3:1, y como nosotros sabíamos que los ácidos en nuestro modelo no funcionaban, dijimos vamos a descarboxilarlo para de alguna forma hacer un extracto con una mayor contenido en CBD y un menor contenido de CBDA. Entonces lo sometimos a altas temperaturas durante 90 minutos y como ustedes ven se descarboxiló bastante, igual nos queda con un poquito de CBDA y logramos aumentar la concentración de CBD y también de THC, porque este es el THCA que como ven se descarboxila y aumenta el THC. Pero como ustedes ven, la composición de cannabinoides es totalmente diferente al producto de uso médico. Entonces lo que hicimos fue estudiar la capacidad neuroprotectora de este extracto y lo que obtuvimos es que es tóxico, porque no está en el MCT, es más tóxico, si comparamos con estos están representados también en contenido equitativo de CBD. ¿Tá? Entonces, están representadas a la misma concentración de CBD y si comparamos, el extracto muestra una mayor neurotoxicidad, a 10 ya es tóxico, y sin embargo, cuando vemos la neuroprotección es un poco más potente porque protege a concentraciones más bajas en CBD comparado con el CBD puro. Sin embargo, es importante destacar que la eficiencia, la eficacia no aumenta. Sí aumenta la potencia, sí aumenta la toxicidad, pero no logramos con ninguna combinación lograr de alguna forma aumentar, llegar a un 80%, a un 90% de nuestra protección. Bueno, entonces para finalizar las conclusiones en cuanto a los fitocannabinoides aislados y purificados, el CBG y el CBD tienen similares actividades neuroprotectoras. Ambas son mejores en atenuar la muerte por rotenona si la comparamos con el peróxido. Esto nos sugiere que tienen diferentes vías de neuroprotección frente a estos dos agentes neurotóxicos. Esta acción neuroprotectora de CBD y CBG es independiente del receptor CB1, es independiente del receptor CB2 y del PPARs, y lo único que mostró cierta reducción en la neuroprotección fue el receptor 5-HT1A para el caso del CBG, y ninguno de los dos ácidos son neuroprotectores. En cuanto a las combinaciones de los fitocannabinoides purificados, no vimos ningún efecto ni sinérgico ni aditivo entre THC y CBD, ni entre THC y CBG. Y en cuanto a los extractos de full spectrum, el epifractam mostró una menor neurotoxicidad comparado con el CBD. Mostró ser neuroprotector y además mostró ser menos tóxico que CBD, lo cual muestra una mayor ventana terapéutica en cuanto a dosis. El Salex tiene similar efecto neuroprotector que el epifractam, sugiriendo que no hay efectos sinérgicos ni aditivos entre los otros compuestos presentes en el epifractam, y que la acción neuroprotectora de este compuesto se debe al CBD. Y sin duda el MST, el vehículo de formulación, tiene un papel clave sobre todo en la no toxicidad causada por el CBD. En cuanto al otro extracto, en donde hay diferentes relaciones entre los cannabinoides y seguramente diferentes composiciones en flavonoides y en terpenos, porque vienen de diferentes plantas, nuestros resultados apoyan la noción de que los extractos sí pueden tener un efecto diferencial comparando con las moléculas aisladas, pero este efecto puede ser tanto más potente, pero también más tóxico. O sea que simplemente lo que hizo fue correr un poquito la ventana terapéutica y no mostró ninguno de los extractos probados una mayor eficacia de la capacidad neuroprotectora comparado con los compuestos aislados. Nada, por último, agradecer a todos los que participaron en este trabajo, el Departamento de Neuroquímica, donde yo trabajo, el Departamento de Neurofarmacología Experimental, donde trabaja Cecilia Scorza, y a PharmaVision, donde está Raquel Pedrade y Edelma, que son los que nos ayudaron, sobre todo para toda la parte del full spectrum, nos dieron los compuestos y los vehículos utilizados para la formulación de estos productos comerciales. Y todas las agencias que apoyaron económicamente y también donaron los productos para poder hacer estos estudios.
Okay, so we have time for questions, and then you are welcome to, as I said before, to ask in language, and we try to help.
Gracias, Caroli, por tu muy buena presentación. Me quedé pensando, cuando describiste los mecanismos de acción que estudiaste y dijiste que el CB1, CB2 no he bloqueado, los antagonistas CB1, CB2 no he bloqueado el efecto del CBD, me quedé pensando porque tu cultivo no tiene las células de glía.
Astrocitos tiene.
Microglia.
Microglia no.
Microglia no. Me quedé pensando porque este mecanismo de protección vía CB2 podría ser vía microglia. O sea, si no tiene la microglia, capaz que el CBD podría no tener este efecto indirecto de protección de las neuronas.
Sí, igual los astrocitos tienen CB2 y las inmunos que mostré cuando presenté el cultivo son hechos en nuestro cultivo. Hay presencia de CB2 en los astrocitos que tienen este cultivo. Y si bien los astrocitos hay pocos, es el 5%, la superficie, el área que tienen estas células frente a los granos, que son células muy chiquitas, es amplia. O sea, hay cantidad elevada de CB2.
Sí, pensé que podría capaz intentar un cultivo organotípico o algo así que
sí,
tal vez
lo pensamos. Sí, son cosas para hacer a futuro. Pero sí, pero hay CB2. Y de hecho, no lo mostré, pero los astrocitos, cuando le ponemos el ipifractán, tienen cambios en GFAP. Digo, no quiere decir que sea por el CB2, puede modificar al astrocito por otro mecanismo. Pero son de las cosas que también estamos estudiando.
Muchas gracias, Carolina, por tu excelente presentación. Considero que la charla es muy interesante porque evalúa no solamente extractos puros de CBD o THC que la literatura ha reportado, sino que también el espectro, el full spectrum que tú hablas de los extractos, abre una puerta para poderlos evaluar. ¿Consideras entonces que en las propiedades que tienen los cannabinoides para otro tipo diferente a neuroprotección deberían estudiarse también de esta manera? ¿Consideras que de acuerdo con tus...?
No, primero que los estudios estos son in vitro. Digo, para ver si realmente esto sucede, hay que pasar un modelo in vivo, sobre todo por la biodisponibilidad. Yo creo que las combinaciones, ya sea en el full spectrum o combinaciones de compuestos aislados, lo que sobre todo va a modificar es la concentración de CBD, que es el compuesto que en mis resultados, o CBG, está mostrando actividad neuroprotectora. Pero no es lo mismo, nosotros lo probamos, cuando yo les nombraba de que vengo de un background de productos naturales de los flavonoides, nosotros tenemos efectos totalmente diferentes con la quercetina, que es un flavonoide aislado, de la marcela, creo que la conocen, porque también está en Brasil, la planta de la marcela, y es totalmente diferente la actividad de la marcela que de la quercetina aislada de la marcela, pero no por el efecto sinérgico de todos los otros compuestos, sino por la concentración a la cual la quercetina logra llegar al cerebro cuando es dada como marcela, como extracto, que cuando es dada aislada. Se concentra mucho más.
Ok, gracias.
Muchas gracias, Carolina, por tu presentación. Me surge una duda, es una duda pequeña. El hecho es que has mostrado que usaste rotenona para inhibir el complejo 1. Entonces, sabemos que las neuronas dependen mucho de la mitocondria como una fuente de energía. En este caso, ¿sabemos o se han hecho pruebas para ver el efecto de estos compuestos en la viabilidad? ¿De qué vía están tirando esta energía, este ATP? Ya que si tengo inhibida la mitocondria, necesitaría alguna otra, y es que se beta oxidación, autofagia. ¿Sabemos algo de eso?
No hemos entrado en ese tema, pero yo creo que en realidad quizás la célula esté metabólicamente igual, más disminuida, y lo que está viendo es un efecto, por otro lado, neuroinflamatorio o de homeostasis del calcio en estas células. Estas células, para que puedan diferenciarse y vivir, necesitan tener en el medio de cultivo cloruro de potasio, necesitan estar despolarizadas, porque si no automáticamente entran en apoptosis. Entonces la homeostasis del calcio es fundamental para la viabilidad de estas células. Entonces yo creo que la protección de estos compuestos vienen más de ese lado. No sé si ahí va la pregunta. No.
Casi, pero bien. No, es que al verla que sobrevive mucho más, pero con una mitocondria inhibida, y entonces la fuente de ATP o de energía, ¿cuál sería la otra posible vía? Pero está por ahí, o sea que está bien. Gracias.
Thanks for the amazing lecture, Dr. Carolina. I don't speak Spanish, so I will try to make the question in English or in Portuguese. I don't know if more suitable. If you already tried to test another phytocannabinoids less studied than CBD, CBG, because I think it could be interesting to apply a preliminary filter, computational filter for screening of direct antioxidant or even other interesting pharmacological properties before testing in culture, because as you mentioned, there is more than 100 phytocannabinoids characterized in cannabis sativa extract, so could be a interesting approach for drug development and to find interesting ligands in this all phytocannabinoid.
Sí. El problema ahí es la obtención de los otros cannabinoides, porque no es, nosotros tenemos la colaboración con Phytoplant Research que nos va donando estos receptores, estos fitocannabinoides, pero no es fácil, nosotros en el laboratorio no hacemos aislamiento y purificación de fitocannabinoides, son los que nos va dando Phytoplant Research. Creo que la mayor limitante es la obtención de los fitocannabinoides aislados y puros. Eso.
The last question here. Carolina, have you tested, has hecho también evaluado el efecto neuroprotector del THC aislado?
Sí, lo mostré. La primera slide, una de las primeras slides que mostré cuando empezamos a estudiar la combinación de THC y CBD y de THC-CBG fue probar la actividad neuroprotectora que protege a concentraciones similares que el CBD y que el THC. Y lo extraño, que no lo mostré, pero lo voy a mostrar. Ahí estoy yendo no, estaba al revés, perdón. Es que nuestra hipótesis era que, ya que CBD y CBG la neuroprotección no estaba dada por CB1 y CB2, juntémoslo con un agonista de CB1 y CB2 que era el THC. Ese sinergismo no nos funcionó, pero también cuando probamos los antagonistas de CB1 y CB2 con el THC, no bloquearon la actividad de THC. O sea que THC es un agonista parcial de CB1, pero en este modelo la actividad de THC tampoco viene dada por CB1 y CB2.
Yo con respecto a los otros cannabinoides que tú preguntabas, es verdad que hay más de 150, pero son las familias, son los metabolitos. Y lo cierto es que son en muy baja concentración, su amplia mayoría, y que no todas las plantas tienen todos. Entonces, eso complejiza mucho más este tipo de investigaciones, porque ¿qué variedad vas a usar para testar qué cannabinoide? ¿Y qué cannabinoide estaría más allá de CBG, CBC, THC, CBD en concentraciones suficientes para evaluar un efecto terapéutico? Pasa lo mismo con los terpenos y terpenoides. No llegan a concentraciones, ni siquiera a la molaridad de cómo están en la planta para que den algún efecto. Entonces, eso complejiza mucho.
There is some approaches in Brazil to study natural products like NUBI database that compiled several natural products in Brazil. And I'm thinking in a similar approach, building a database for phytocannabinoids and test this database in virtual screening approach to select more possibly active compounds for interesting pharmacological properties. It's possible to construct virtual screening models to predict antioxidant activity, target activity from CB1, CB2, I don't know. Also phenotypic assays for neurotogenic activity, so could be an interesting approach before test it in cells or in vivo experiments. So I don't know, could be interesting in a drug development timeline.
Actually, because we have time, and I overlooked, but we actually have, okay, a little bit more here. So I go in this way, if that's okay.
Muchas gracias por la presentación, me ha parecido muy interesante y he aprendido mucho porque nosotros trabajamos con cannabinoides pero siempre sintéticos y siempre desde un punto de vista muy farmacológico lo que intentamos es buscar lo que sea más específico de receptor y todo, ¿no? Entonces Mi pregunta es, estos extractos, la traslación que esto puede tener en un futuro, al in vivo y luego a la clínica, ¿no sería un poco complicado? Porque nosotros ya vemos en la farmacología como más pura que el antagonista CB2, luego hacemos ensayos de binding, de actividad, y no vemos que no está en CB2, o el agonista específico CB1, vemos que no está en CB1. En estos extractos que tienes tantas cosas, ¿no? Y algunas que ni siquiera son CB1 ni CB2, no sé, simplemente como reflexión, ¿no es difícil de trasladar a la clínica?
Y encima, además, los resultados que obtuve apuntan más hacia que el efecto está dado por el CBD, que justamente apoyando lo que vos decís, los otros compuestos pueden llegar a potenciar, pero también están dando un efecto más tóxico. Entonces, obviamente que en la clínica, para mí, va a complejizar mucho más que usar el CBD solo. Lo que yo esperaba era, aunque sea combinaciones entre cannabinoides puros y aislados, potencie la acción o sinergia, o sea, aunque sea se adicionen y no hay efecto. No me queda claro si es porque están actuando por el mismo mecanismo de acción o no lo sé.
Muy interesante de todas maneras. Muchas gracias.
Gracias.
Carolina, gracias por la charla, muy linda. No, me quedé pensando en el vehículo, en las pruebas que hiciste con el vehículo MST y CBD, y que aumentaba la neuroprotección o mejoraba. Si habían hecho o si se podrán hacer pruebas con CBG a ver si se puede vehiculizar también en MST y no sé si ya lo probaste o
no lo probamos, pero sí se puede vehiculizarse. Y lo que no probamos tampoco, perdón, te corté.
No, si pensaban hacerlo o si ya lo habían hecho, si tenían alguna información.
No, sí pensamos hacerlo. Y sobre todo lo que también tenemos que hacer es hacer las combinaciones en MST, porque capaz que hacer las combinaciones, la biodisponibilidad de estos compuestos cambie en el contacto con la célula y ahí capaz que podamos ver algún efecto. Pero sí, me está todo por hacerse.
Many thanks for some wonderful presentation. My question relates to the mechanisms of these effects. I was wondering if they could involve antioxidative effects, because both CBD and THC, I don't know about CBD, but I imagine it would be the same, have potent antioxidative properties by their molecular structure. And on top of that, I remember reading that CBD is actually has a more potent antioxidative effect than THC. And then I was struck by your data, because if I'm not wrong, when you showed the isolate effects, CBD was actually more potent than THC.
Sure.
So I was wondering if these antioxidative effects could be playing an important role in these neuroprotective effects.
Estoy de acuerdo que la actividad antioxidante debe tener que ver, pero no, si fuera realmente la actividad antioxidante tendría que proteger mejor en el modelo contra el peróxido de hidrógeno y no contra la rotenona. Nosotros la quercetina, vuelvo a nombrar la quercetina, que es un potente antioxidante, excelente antioxidante. Nosotros hicimos ensayos de capacidad antioxidante por el ABTS y la quercetina, CBD y CBG tienen capacidad antioxidante similar, la quercetina es más potente, pero la quercetina en el modelo de peróxido funciona divino, protege a las 24 horas de un 80% y en el modelo de rotenona no protege. Estos compuestos protegen poco en el modelo de peróxido y sí protegen en el modelo frente a la rotenona. Entonces, debe tener que ver, pero no explica todo. Sí. Son necesarios. Sí. Sí.
Muchas gracias por su presentación. Mi pregunta igual va relacionada con el modelo con rotenona, que va dirigido al complejo 1 de la mitocondria. En este sentido, el receptor CB1 mitocondrial también está relacionado con el complejo 1 de la mitocondria. Entonces, pensé si se ha contemplado evaluar este receptor mitocondrial en la protección en el modelo con rotenona.
Sí, lo teníamos planteado, pero desde el momento que el antagonista entra a la célula, también nos está antagonizando el CB1 mitocondrial y la actividad neuroprotectora no se ve cuando está antagonizando ese receptor. ¿Me explico? Una cosa que no sé quién fue una estudiante que habló ayer, que habló sobre la biogénesis de mitocondria a través del antagonismo de CB1 mitocondrial, que yo me quedé pensando que capaz que sí podría estar haciendo ese efecto, que haya más mitocondria y por eso no esté afectado, de alguna forma al ver más mitocondrias, el daño por la rotenona no sea tan brusco.
Thank you very much, Dra. Carolina, for this very nice talk.
Gracias a ustedes.
So just before we go to lunch, we have short reminders from Ismael, and then just before I head on to him, I would just like to say we are very happy to see this very fruitful interaction from everyone, and that's exactly how we wished it should be. So
yeah, we should keep the discussions lively like we did today, so well done. There will be a beer on the dinner on Wednesday. Second thing is you guys can go for lunch to the philosophy school. I'm sure Carlos or Aníbal here that are locals won't mind to walk with you guys through to philosophy school. And I'm going to put the names here of the students on the slide before you guys go so the PIs can start to go. And the students just give me 5 seconds for the distribution between the two modules. So remember the modules are, they are not compulsory. You can either choose to do them or not. But it's good to know at least how many people we're going to have on the module. So we're going to provide you a distribution according to the survey that we did, or what was the interest, and then you can split between module 1 and 2 between today and tomorrow. Okay? So module 1 will be here in the School of Medicine. We will meet downstairs in the main door. The other module will be in the School of Biology. Someone, Javier Palazuelo, Santana Aguado, will grab you on the food where we're going to have the food because it's closer. And then after that, at 7, we have the medical chemistry session here on this room. Okay? In this room. That's it. Thank you very much.